ladies and gentlemen, on behalf of Academy of Europaea, I would like to open the Class C meeting. Welcome, everybody. I am very happy to share the session with the President, Sir Kluting, and also with Eva Kondroshi, who is the Chairman of Class C. Myself is Peter Hegyi. I am member of the Academy of Europaea from 2018. So let's start with the President welcome. So I would like to ask Sir Kluting to give his opening speech.
A very warm welcome to all people attending this class meeting of the Life Sciences class. And of course, in particular, a warm welcome to the new members elected in 2020. We are a young academy, and I cannot resist by showing you this slide, which is the slide of the foundation meeting in 1988 in Cambridge. And what is important to mention here, that this was a completely bottom-up initiative. A group of scientists from all over Europe realized there was a strong need for a pan-European academy, particularly at a time of major changes in the political landscape in Europe. And I'm very happy that amongst the founders of the Academy, we had Ole Pedersen, presently a vice president of our Academy and the director of our Cardiff hub. And again, this type of initiatives are important. In the same spirit, a group of people, a little bit smaller in size, took some years later the initiative to create a European Research Council. Again, nobody told them to do so, but it was their initiative, and they realized it was highly needed to have a European Research Council as part, let's say, of the framework program of the European Commission. Today, we have close to 5,000 members from all fields of sciences, including the social sciences and humanities. But you can see also that life sciences is a big component of our membership. And we have elected recently 361 scholars to join us. So we are quite stable in terms of our composition, but we are also quite stable in terms of, let's say, our membership at large. The mission of the Academy is basically to promote wider appreciation of the value of European scholarship and research, to make recommendations to national governments and international agencies, including the European Commission, concerning matters affecting science, scholarship and academic life in Europe, and I will give you later on some examples, and to encourage interdisciplinary and international research in all areas of learning, particularly in relation to European issues. And of course, that is directly coupled to what is listed here under bullet point 2, because the issues that we are called upon for science advice or policy are by their nature of interdisciplinary character. What is also important is, of course, that we see as our mission to identify topics of trans-European importance to science and scholarship and propose appropriate action to ensure that these issues are adequately studied. Our strategic priorities are directly linked, of course, to our mission. First of all, to recognize and support the development of individual scholarship and scholars. It's, after all about people. People make the difference. Individual scientists makes the difference. Second, of course, to deliver collective and independent excellence in expertise and activities. Create beneficial collaborations and partnerships. Because we are not a standalone organization, but we work in close partnership with other components of the research and education and innovation landscape, to improve visibility, impact and trust, and to broaden the support base. Very few words about our governance. The Board of Trustees is composed of people with different backgrounds from all over Europe. You see here a few pictures from some of its members. And in the next slide, you see some other members of our board. And also like to mention here that the class chairs are member of the board. And you can see from the different examples listed here that most of these people are also serving in other capacities for the scientific community, including, for example, Professor Eva Kondroshi, who is member of the group of chief scientific advisors to the European Commission, a former vice president of ESC. Professor Eystein Janssen, director of the Bergen Knowledge Hub, is currently member of the Scientific Council of the ERC. Then we have also Professor Don Dingwall. He's a former Secretary General of the ERC, and of course, very important also to mention here, Dr. David Coates, our Executive Secretary, who is not a trustee, but is of course playing a very important role in the life of our Academy. Your class consists of 5 academic disciplinary sections, and each of them has a section chair, vice chair, and a number of appointed and elected committee members. And what is, of course, very important is to realize that, of course, for us, it's important that you feel home in your section and that you feel home in your class. And for that context, of course, it's important that you realize that all these people in the different section committees are very eager to establish a personal contact with you. Let me move on. And stress that supporting excellence and scholarship is key in our mission. An example, the Erasmus Medal, awarded annually for sustained scientific excellence. 2020, Roger Penrose, Nobel laureate, received the Erasmus Medal for his groundbreaking research in mathematics. 2021, Philippe Adjunct received the Erasmus Medal for his pioneering work on economics. In addition, we have the Hypatia Prize, sponsored by the city of Barcelona for scientific achievement in all fields of sciences, a prize that goes also along with 30,000 euro. And we have the Eva Kondroshi Prize, a prize in your class, a biennial prize for excellence in plant sciences. In addition, we support excellent activities, for example, the Hercules Group of Academia Europaea, a working group on higher education, research and culture in European society, chaired by Lars Engel from Uppsala, which is basically an Academia Europaea wide group that organizes international conferences on topics relevant to the practice of research and higher education in a transdisciplinary and international context. They publish symposia volumes and reports in the European Review, our own journal, published by Cambridge University Press. In addition, of course, we are very pleased also by class activities, such as the ones by Class C, your class, supporting excellence. And in this context, of course, I cannot resist to show you some examples. The future of translational medicine, for example, but also the basically topic dealt with in the Vienna Congress in 2021, Back to the Future, scenarios for the world with and after Corona. So again, plenty of opportunities also for our new members to be involved in activities that make a difference. What is also an important point is to mention that we are a pan-European academy. That means that in addition to our headquarters in London, that we are present in all over Europe through our regional knowledge hubs, each with a thematic focus, but also with a regional focus. And of course, I like to mention in the context of this class meeting, in particular the Cardiff hub, very active hub for us, with Ole Pedersen as academic director. Next. All these hubs are powerhouses for activities, and they're either based at universities or at academies. But please realize that your involvement in the functioning of the hubs is also an important issue, and that also these hubs basically can be very useful to you in order to get activities off the ground, workshops and other things, or bringing people in your area together for Academia Europaea meetings in addition to what we do in the annual conference in Barcelona. The Graz Information Center is also an important issue to mention here, because you have been dealing with them recently, because they are responsible for the development and application of the nomination system and the membership administration and registration. And these are the people here on the slide who make the difference here. They run it, and so they are always very helpful to our members for any questions that are concerning your membership and other issues. So don't hesitate to contact them. They're very responsive. Another important thing to mention here is the Young Academy of Europe. This is an initiative of some years ago by a group of starting grantees of ERC, who realized that with receiving such grants, it also means a certain responsibility. A certain responsibility also to take an active part in taking the future of their own generation on their shoulder. And I'm very happy with the close affiliation of the Young Academy of Europe with our own academy, which is also reflected in the joint organization of our annual conference. And again, please do not hesitate to nominate members for membership of the Young Academy of Europe, because we have an arrangement with the Young Academy of Europe that our members can do so. So if you basically are aware of early career outstanding scientists, and that you say, well, this will be perfect people for a Young Academy of Europe, please nominate. Another recent development is the establishment by the European Commission of a scientific advice mechanism. The European Commission feels the need to ask for science advice for policy on issues which are important to deal with in legislation, but also in terms of regulations. And the questions basically come from the Commission at large. So not just from, let's say, the Commissioner for Science, Research and Education and Innovation, but from the Commission at large. The system has two components. First, a compact group of outstanding scientists, which is called the Group of Chief Scientific Advisors. And here one of the members is Professor Eva Kondroshi, one of our board members, and SAPEA, which stands for a consortium of the scientific academies in Europe. And well, here you see a little bit more background on SAPEA. The acronym stands for Science Advice for Policy by European Academies. And it's a consortium of 5 partners. The partners are Academia Europaea, that's a pan-European academy, but also federations of national academies. Federations of national academies such as Europaea, which is a federation of engineering academies in Europe. ALEA, well known to many of you, Federation of National Academies in Europe. And FEAM, a Federation of European Academies of Medicine. And in addition to this, ESAC, the European Academy Science Advisory Council, is also a member of the consortium. And we, as Academia Europaea, are a member of ESAC itself. Why did the European Commission go for this consortium of academies as part of the scientific advice mechanism? Well, the answer can be very short, because we are providing independent advice. We are not there, let's say, for a particular interest, a lobby group or whatever. We are there as representing the scientific community individuals. And we serve here by providing basically knowledge where the science stands on topics which have to be dealt with by the European Commission. The products are basically science, are evidence review reports. But before, let's say, I come to that, I like to mention here very briefly for what is the essence of what we do as SAPEA. But first of all, to provide the best possible scientific advice for EU policy. Second, and this is a very important spin-off, connect academies and networks across Europe, because that is highly needed. We have to connect what's going on the national level with the pan-European level. And that's exactly what SAPEA does. And third, promote the role of science in policy making, increase transparency and public confidence in EU policy making. There are, of course, a couple of challenges in this. First of all, how can we strengthen the dialogue between science and policy? it takes two to tango. And it's all a matter of trust. But it has developed in a very good way that we see basically that we have recipients for our report in terms of that the European Commission is very eager to receive our reports. And that's of course important in this mechanism that basically something is done with our reports. Second, how can we develop science for policy relatively fast? And because some of the issues are pretty urgent, and at the same time we have to ensure highest quality. And again, this is a major point for our academies to basically secure this quality, but also secure the issue of independent advice, to select experts that have no conflict of interest, so to say. Just a few words about the reports and about the products. The products are called evidence review reports, and they are the base for the scientific opinions produced by the group of chief scientific advisors. The abbreviation of these evidence review reports are ERRs. And if you look basically to the topics covered, you see it's quite a broad range, ranging from cybersecurity, agricultural biotechnology, food from the oceans, novel carbon capture and utilization technologies, plant protection products, microplastics, transforming the future of aging, making sense of science, sustainable food system, health effects of climate change, and biodegradability of plastics. Well, it's very clear that you recognize a lot of topics where you can already identify how important the impact from members of our class of life sciences will be. But in addition, you also realize that most of these topics also require an input from other fields, including the exact sciences and social sciences and humanities. In the next slide, we zoom in on a number of ERRs where basically Academia Europaea was the lead academy, because we are quite active in this consortium. And basically the first one, Food from the Oceans, established the model for all the others. The other one, Making Sense of Science, very influential report from policy under conditions of complexity and uncertainties. Biodegradability of plastics in the open environment. They organized recently an event in the European Parliament, chaired by Ole Pedersen. And in addition, the Cardiff hub of Academia Europaea led the systematic review on the sustainable food system for the European Union. What's going on today is illustrating, let's say, the impact and the importance of the topics that we deal with. Well, for example, systematic approach to the energy transition, very important topic in the context of the Green Deal of the European Commission. Academia Europaea led the mapping of the policy landscape for this report. Then crisis resilience, also very important in the context of COVID, and how to deal with the consequences, let's say, of crisis situations, and how to be better prepared. Academia Europaea has been leading the systematic literature review, and the same is the case for cancer screening, another very important topic, and a topic also of one of the missions of the European Commission. Also here, Academia Europaea is leading the systematic literature review. So let me conclude again by extending a warm welcome to our new members of Academia Europaea, and hoping that you draw much inspiration as a new member. Academia Europaea is all about people. They make the difference for our academy. We count on your active involvement in our activities. Membership of our academy is more than an honor. It's more than just to put something in your CV. Your active involvement is very important for us, and we need you also to engage in activities in the broader scope of science and society, such as the Science for Policy advice. So please enjoy the rest of this session and our plenary conference on 2021 October. And of course, I cannot resist by displaying here a few program highlights, including the Erasmus Medal lectures and the Academia Europaea Gold Medal lecture. And the first case, of course, as I mentioned, Erasmus Medal lecture is given by Roger Penrose. The Academia Europaea Gold Medal lecture is given by Ole Pedersen. And then we have two Balzan Prize lectures. by respectively Susan Trumbore and Quinton Skinner. But in addition, we have also two very exciting talks on respectively volcanism and the mysteries of memory. And last but not least, also sessions organized by the Young Academy of Europe, including the session centered on the André Mischke Prize for Science and Policy, and a panel debate organized by the Young Academy of Europe on redefining rewards and recognitions for European scholars. So again, it's great to have you with us, and I wish you again much inspiration through your active membership. Thanks again.
So thank you very much for the very inspirational welcome, and now I would like to ask Eva Kondroshi to present her welcome at class C meeting.
Good afternoon and very warm welcome to all of you, and particularly the new member of Class C. My name is Eva Kondroshi. I am the interim class chair of Class C. In the coming few minutes, I would like to give you a brief introduction on the structure of the life sciences and also about the statistics of the membership. So in Class C, we have 5 sections: biochemistry and molecular biology, cell and developmental biology, physiology and neurosciences, organismic and evolutionary biology, and clinical and veterinary science sections. We have altogether 1,255 members. Here you can see the memberships in the different sections, and also that we have 17% female members only. If we look the members distribution by country, we have members from 42 countries, and we can see here the predominance from UK, France, Germany, Netherlands, and Italy. In the biochemistry and molecular biology sections, I am the section chair, and here are members of the section committee. We have members from 33 countries. Altogether, we have 346 members, and 18.5% are the females. We have many members from Western Europe, particularly from France, United Kingdom, and Germany, Italy, and Switzerland. And from the Central and Eastern European countries, we have quite a broad representation and relatively high numbers from Hungary. The cell and developmental biology section is chaired by Crisanto Gutierrez, and here are members of the section committee. This section covers cells and developmental biology, genetics, immunology, and microbiology. There are 231 members, 17.7% are the female members, and again we can see the dominance of the Western European countries in the membership, particularly UK, Germany, France, Italy, Netherlands. The physiology and neuroscience section is chaired by Balázs Gulyás. This section covers broad fields: physiology and biophysics, neuroscience, clinical science, pharmacology, and toxicology. Here are members of the section committee, and this section comprises 428 members. 17.5% are only the female. Again, many numbers from the United Kingdom, followed by Germany, France, Italy, Netherlands, and Sweden. From Eastern Europe, the only significant number are from Hungary. The organismic and evolutionary biology section is chaired by Christophe Thibault. Here are members of the section committee. This section covers population biology, environmental biology, biosystematics, evolutionary biology. This is a small section, 126 members, 14.3% are the female, and most members are from the United Kingdom, France, and Germany. The Clinical and Veterinary Science section is chaired by Stefan Holgate. Here are members of the section committee, and this is a newly created section with few members so far, 124, but this is under creation and development. Many members from the United Kingdom and Netherlands, and so far from 24 countries. So in the life sciences, we have many challenges in the class. We have to improve gender balance and geographical diversity. It will be necessary probably to make a restructuring of Class C to better reflect all fields of life sciences, particularly the emerging areas and multidisciplinary research. We have to better exploit the knowledge and expertise of Class C when scientific advice is needed. We have to make Academia Europaea and Class C more visible and known for the public. We have to promote science and to be involved in mentoring programs. So for all these activities and other future activities, we really need your active participation. And now we come to the scientific talk, the keynote talk, which will be given by Katalin Kariko on the development of messenger RNA for therapy. Kati was elected to Academia Europaea in 2020, and she belongs to the biochemistry and molecular biology sections. I think I do not need to introduce you. Her scientific achievements are unquestionable. Her invention, the development of messenger RNA technology for therapy, will make certainly a revolution in medical science, giving also chance for recovery from some presently untreatable diseases. The messenger RNA vaccine development against COVID-19 saved already millions of lives and will help in the future to overcome other pandemics as well. Kati can be a role model for all of us. Her scientific achievements were reached by her strong convictions, determination, commitment, perseverance, stubbornness, and very hard work. I know Kati since the 80s, when we worked together on the introduction of DNA to mammalian cells with liposomes in the Biological Research Center in Szeged. I know that for her, the greatest joy comes from the excitement of research, as well as the awareness that her work has contributed to the creation of an important technology in therapies. Kati got several prizes, or many prizes during the last months this year, and already some prizes are already awarded, which will be given only next week. Here are a few examples. These are very prestigious prizes, and I hope very much that I can introduce her as a new Nobel Prize winner, which did not happen, unfortunately, but I am very proud that she was elected to Academia Europaea already last year, and this was really an international recognition for her. Kati wanted to come to this meeting and give her talk here. However, and we have to congratulate her because she received also the Princess of Asturias Award 2021. She is presently in Spain, but already taking part of this ceremony. And now I will give the floor to Kati to give her speech on development of messenger RNA for therapy, which happened not light speed. Thank you very much.
Dear colleagues, today I will talk about development of mRNA for therapy. I worked on this field about 30 years, and I can tell you, it did not happen at the light speed. The messenger RNA was discovered 60 years ago, 1961, and these are the main timeline for how in the last 60 years messenger RNA was developed into a therapy. 1978, when mRNA was first delivered to mammalian cells and the encoded protein has been shown. 1984, mRNA was synthesized in vitro. 1990, first when mRNA was injected in vivo in a mammalian, in the mice. And 2000, when the in vitro transcribed RNA found to be inflammatory. 2005, we had a solution and made non-immunogenic RNA. 2017, first time has been shown that LNP formulated nucleoside modified RNA is a good vaccine in preclinical studies. And by 2021, 60 years after mRNA discovery, we had an FDA approved vaccine. So 1958 was one of the experiments was performed scientists who were looking for find out how the information from the chromosome, which is present in the nuclei in the case of eukaryotic cells, how this information is coming to the cytoplasm to make protein on the ribosome. So they tried to investigate and found that there are some RNA is associated with ribosome, and they thought that maybe that is the RNA which the messenger RNA which they were looking for exists. But this very interesting experiment, Russian scientists demonstrated that the ribosomal RNA cannot be the elusive messenger RNA, because they had different bacteria and found that the DNA has a very different GC content, from low GC content to high GC content. But when they looked at the RNA in this, which is mostly was ribosomal RNA, they found that the composition was identical. So the RNA associated with the ribosome was the ribosomal RNA, and it was not the messenger RNA. It was 1961 when two papers reported existence of the mRNA, both of them interestingly and importantly saying that it was a very unstable intermediate. And subsequently, demonstrated that in the case of eukaryotic cells, the RNA is leaving the nuclei, getting to the cytoplasm, where on the ribosome it is translated and the protein synthesis proceeds. Of course, discovering the RNA was not made it possible that the RNA could be made. So at the beginning, scientists just could isolate RNA. And they did this 1978, this very important experiment, when they isolated globin RNA from reticulocyte, rabbit reticulocyte, and they wrapped this isolated RNA into liposome. This liposome was made how Papahangiopoulos described in the mid-70s, and large unilamellar liposome generated with the film methods. And the wrapped RNA was delivered to mammalian cells, in this case primary lymphocytes, and demonstrated that the coded protein was made inside these cells. This was published in two papers, again, both of them in Nature back-to-back, and articles. And so this was the first time, 1978, when messenger RNA was delivered inside the mammalian cells and the encoded protein had been demonstrated. While these experiments were ongoing, we were reading about it, and we tried to do something similar. We could not isolate RNA, so instead we used plasmid DNA, and again, using the same method described by Papahangiopoulos, we put in the large unilamellar liposomes, and we delivered the plasmid DNA inside mammalian cells and demonstrated the encoded protein was made. In 1984 was an important milestone here, because first time in vitro transcribed RNA was generated. Douglas Melton and Paul Krieg at Harvard University used the phage RNA polymerase. Scientists before them, they tried different kind of RNA polymerases to make in vitro RNA. Those were bacterial RNA polymerases. But in the case of phage polymerase, it was high fidelity. So and also the RNA was generated from needed a very short promoter. They used sp6 phage RNA polymerase and generated RNA, which coded for human beta interferon. It had 5' UTR, 3' UTR, poly-A tail, which was encoded in the plasmid, and they also had a cap structure on the 5' end, which they did with capping enzymes. They injected the generated RNA into frog oocytes, which are light cells, and they injected several of these and then incubated overnight, collected the medium, and they demonstrated that the protein was present in the culture medium. The interferon was functional, inhibited viral replication. So this was the first experiment that in vitro transcribed RNA was translated inside a cell, which generated functional protein. The next major step was when in vitro transcribed RNA was injected in mice. This was a very simple experiment, but very important for all of us who were on the field, because demonstrated that when the RNA was injected in vivo in an animal, in the muscle of the mice, that the encoded protein is made. The RNA was luciferase encoding RNA, and this was published by John Wolfe and colleagues. This encouraged many scientists all over the world, and here in the first part of the 1990s, these scientists tested out messenger RNA for different applications. Most importantly, 1992, vasopressin mRNA was injected to the brain of a rat, which was an animal model for a disease. And Floyd Bloom and colleagues at the Scrip, they demonstrated that they could treat this Brattlevoro rats, which were diabetic, after injecting the vasopressin mRNA. 1993 was the first paper when described messenger RNA used as a vaccine. These were French scientists, Martin and colleagues, Meulian and others, and they delivered the messenger RNA in liposome and demonstrated that the influenza-specific protein was made and it was worked as a vaccine. Similarly, performed an experiment in Karolinska by Lilleström and colleagues, and they used a self-amplifying RNA and injected the naked RNA to the animals. And later on in 95, 96, when first time messenger RNA was used as a cancer vaccine. The first experiment, Conry, Curiel and colleagues injected naked RNA to the animals and demonstrated biological effect. In 96, Eli Gilboa at Duke University, he used in vitro transcribed RNA and exposed, isolated, and generated human dendritic cells and treated those human cells with in vitro transcribed RNA. He went on and made the first mRNA company, Merix, which became later Argos, and their clinical trial went all the way to phase 3. And in the later years, actually, most of the study was focused on cancer vaccine and not infectious disease vaccine. While these colleagues were working all over the world, I was also working on a messenger RNA. In 1989, I started to make a messenger RNA coding for urokinase receptor. I worked at the University of Pennsylvania, and my colleague at cardiology was interested in this receptor, so I cloned and made the RNA. We use T7 RNA polymerase, and actually today also this RNA polymerase is used to generate the vaccine for, generate the RNA for vaccines. We use a cap analog. In that case, it was a simple non-reversible cap analog was added, so the capping was done transcriptionally. The RNA was lipofectin complex and transfected into mammalian cells. For us, it was a very important experiment because urokinase receptor function required all of the post-translational modification and processing of the carboxy terminal, and all of these modification was required for biological function. So when we demonstrated that the receptor was functional on the cell surface, we knew that the mRNA will be good for something. It's not even in vivo, but at least treating cells ex vivo. Of course, all of these years, I was doing different kind of mRNA to see that which kind of clinical application it would be feasible, but came to the realization in 2000 that the RNA I'm making is inflammatory. I met Drew Weissman in 1997 when he joined to the University of Pennsylvania as a faculty, and he was interested to generate HIV-specific vaccine. So I made a GEG mRNA for him, and when he tested out on human dendritic cells, he thought that it was a great vaccine, that the RNA could be a great vaccine because it coded for a protein which made the cell made a lot, so the RNA was a good source for the antigen production, and all kind of activation markers were up on the dendritic cells, and inflammatory cytokines were also made, which was good news for a vaccine producer, but not for me, who wanted to develop mRNA for therapy. So we were thinking about this, why the single-stranded messenger RNA is activating the dendritic cells and generating these inflammatory cytokines. At that point, in 2000, it was not known that even the double-stranded RNA, how it can induce interferon. So none of the RNA sensors were known, but it was known that double-stranded RNA is what can induce interferon. What we have done is thinking that whether the RNA, which is inside the cell, maybe is also immunogenic when it is getting out and getting to the immune cells from outside and maybe recognized as an invader. So to test out, we isolated the RNA from different compartments of the cells and transfected into the monocyte-derived dendritic cells and measured TNF-alpha. What we found is that in vitro transcribed RNA was the most immunogenic, induced the most TNF-alpha, whereas those which were isolated from the mammalian cells were less immunogenic. But surprisingly, the tRNA was not immunogenic at all, didn't induce any TNF-alpha. Knowing that tRNA contains a lot of modified nucleosides, here shown in the pink and red, it's about like 25% of the total nuclear content is modified. We were thinking that maybe modification is important and responsible for suppressing immunogenicity of the RNA. Of course, there are so many different kind of modification is known, and the enzymes which incorporate these modification in the RNA, which is always made from the 4 basic nucleotides, it was not known. And so how we could introduce all of these modifications? So we wondered and then finally decided that we have to purchase nucleoside modified derivatives, the triphosphates, and then incorporate those during transcription. So we purchased these 10 nucleotide triphosphate, modified nucleoside containing and generated RNA. Not all of them was accepted substrate for the T7 RNA polymerase. So, but we could generate 5 of those. And then we started to analyze whether they are immunogenic or not. At that point, we made toll-like receptor overexpressing cell lines because by 2003, we knew that toll-like receptor 3 is activated by double-stranded RNA, toll-like receptor 9 with DNA. So we thought that maybe some of those other toll-like receptors responding to messenger RNA. So when we tested out the RNA which was generated with conventionally was inducing quite high level of IL-8. But when we modified and incorporated this modified nucleoside, that RNA was not immunogenic, didn't induce IL-8, suggesting that Tol7 and Tol8 was responding to this RNA we have made. Subsequently, we tested out the same set of RNA now that is not monocyte-derived dendritic cells, but isolated from human blood. It was a little bit different result because at that time only modified uridine-containing RNA were the only one that did not induce any TNF-alpha in the dendritic cells. So it seems that nature selected uridine as the nucleoside recognized by the human immune cells, and when these are modified in any way, methylated in the 5' position or thio at the 2 position, then or using as pseudouridine, whereas uracil is differently linked to the ribose, then those are not activating this toll-like receptor 7 and toll-like receptor 8. We learned this more 10 years later when the crystal structure of these two toll-like receptors were published and demonstrated that even the uridine, the nucleoside itself, is sufficient to activate toll-like receptor 8. And it perfectly fit to the groove here in the crystal structure shown. But when it is modified, then this activation will not occur. Of course, we wanted to do whether it is a vaccine or whether it is a therapeutic RNA, but both cases we needed the RNA to be translatable. And surprisingly, we found that when pseudouridine was present in the mRNA, translation was 10 times higher than the conventional U-containing RNA. And it was both in culture cells or primary cells. When we injected to animals, now it was complex with transit, we found that when the RNA was nucleoside modified, we could detect the protein, the encoded protein, much longer for a couple of days in the animal blood. This was a very small amount, 0.1 microgram RNA was injected to the animal, coded for erythropoietin, which generate red blood cells. What was important that when we checked out the interferon in the blood of these animals, we found that the nucleoside modified RNA did not induce interferon, whereas the U-containing RNA did induce. It was important to see that the RNA coded for a functional protein, as the erythropoietin increased the hematocrit value of the animals. When we weekly injected very low dose, 0.1 microgram EPO mRNA to the mice, we could maintain high level of hematocrit for a couple of weeks. Subsequently, a lot of modification improvement was performed on the in vitro transcribed RNA, the codon optimization, optimization of the untranslated regions, purification, and introduction of the modified nucleosides, so that as we perform these improvements, we could generate more protein from the RNA, and the translation lasted much longer. It was an important improvement was when we introduced the Cap1 structure, which made a much higher level of translation from the RNA. Considering the codon optimization, I just mentioned here one example for that. When we try to eliminate the uridine, which is inflammatory, we exchange it for pseudouridine, and so it made the RNA non-immunogenic. Others selected an approach to replace the codons and made it GC-rich, the sequences, which eliminated many of the uridine from the construct, but we have to know that half of the amino acid still needs at least one uridine present in the codon. So if you are using this GC-rich codon optimized sequence, the immunogenicity of RNA always depends on what is the amino acid composition of the protein, and how are those amino acid which requires uridine is present in the protein. Now that we are 2021 and beyond, we already know that nucleoside-modified mRNA is in human trial. One of them, of course, the vaccine is approved, but the clinical trials is ongoing. The most advanced was for acute disease. VEGF-A mRNA was used for treating heart failure, and this is in phase 2 trial now. Patient who had bypass surgery get injection of VEGF-A mRNA, nucleoside-modified RNA, to their heart, and the same VEGF-A mRNA was used and still used in a patient who had a necrotic wound, diabetic patient, and demonstrated accelerated wound healing in their case. Modified nucleoside-modified RNA used as a cancer vaccine, and also used in intratumorally injecting mRNA coding for different cytokines, and those would transfer the cold tumor to hot tumor. For infectious disease, nucleoside-modified RNA coding for monoclonal antibodies are used as a passive immunization, and some clinical trial is ongoing on that. And for genetic disease, just recently we learned about Intellia results, where they used nucleoside-modified RNA coding for Cas9 and then performed genome editing in the liver cells of a patient. Preclinical studies are going on in vaccination against different diseases. Tolerization for autoimmune disease was published by BioNTech, where multiple sclerosis model animals were treated with mRNA, nucleoside-modified RNA coding for autoantigen, and genome editing, as well as treating edema with VEGF-C mRNA to generate new lymphoid vessel is ongoing. So this is the therapeutic use, and of course, very important is how the vaccine was developed with the nucleoside-modified RNA. The 2012, when lipid nanoparticle was used as a vehicle to deliver modified mRNA to cells and then animals. The LNP is containing 4 different lipids: the ionizable lipid, helper lipids, pegylated lipid, and cholesterol. Each has its own role. And this LNP formulated mRNA was first used by Norbert Pardy and colleagues Drew Weissman laboratory, where they generated messenger RNA encoding for the envelope of the Zika viral construct. This was one methyl pseudouridine containing RNA, and it was formulated by Acuitas. And this small particle were used to inject to animals. The important was that when monkeys were injected, and just using 50 microgram of RNA formulated in LNP was already providing protection for the monkeys. That was surprising, because prior to that, usually the much higher dose were needed, let's say when DNA was used as a vaccine for larger animals. And here it seemed that like 50 microgram protecting the monkeys, and that was protective for mice as well. So it didn't have to scale up unproportionally or proportionally for the larger animals. So when the animals were challenged with Zika, then it was demonstrated that the virus could not proliferate in the monkeys. So this is how we reached this point where the vaccine was developed, and as everybody knows already, that in 2020, last year, when sequence was available in January for the SARS-CoV-2, then companies like BioNTech, Pfizer, and Mrna started to make a vaccine against this coronavirus. And as we all learned, in 2020, the vaccine was produced, tested, and already get emergency use approval. So this was the most important part, that in 60 years from the discovering the messenger RNA, we reached a point in 2021 when the BioNTech Pfizer vaccine was fully approved by FDA. So there are a lot of people worked on this project from the day one, from discovering the RNA, generating in vitro transcribed RNA, testing out in different model systems, improving the RNA, improving the LNP, and this is where we are today. And for the future will be very likely further improvement, especially the formulation, which will help to in vivo target different tissues and solve different kind of medical question and medical treating a patient with different kind of disease which was not available up until now without messenger RNA. So now I thank you for your attention.
So, Kati, thank you very much for your excellent talk. It was really great. And I have to correct myself because I told at the beginning that you are not available at our meeting today. But it shows that we can count on you even if you have a tremendous program and you will have time to join us. So very warm welcome, Kati, that you are here. And if anybody would have any questions, Kati will be available to answer these questions.
Thank you very much. Actually, I am here
Hello.
in Spain. Hello. I am here in Spain, in Oviedo.
Oviedo. And how is the ceremony ongoing?
That will be on Friday, but yesterday the welcoming was so beautiful, bagpipe playing and cheering the residents of Oviedo that you know I just had to cry. I was just so emotional. And today I visited a school. And children were so grateful, and so I enjoy every minute of being here in Spain.
That's great to hear. We are really very happy that you managed to come and we can see you here and interact. I see that there is somebody who would like to ask a question. This is Fatima, please.
Hello, Katalin. Very nice to meet you, and very nice to meet you all. My name is Fatima Gebauer. I'm from the CRG in Barcelona, and I work with RNA as well in the RNA field. And I guess one of the main challenges of the RNA vaccines is to really, to really overcome the transient nature of the expression of protein, right? Because at some point the mRNA will be degraded and then that's it, right? So how do you envision that this will significantly improve in the future? Because it appears as if the major, say, features of the mRNA have already been targeted, including, codon optimality, CAP, UTRs, whatever. So from now on, where do you see the major development will be?
I mean, for the vaccine, we don't want the RNA to be around too long because that could be not helpful. So for the vaccine, it's good that the RNA is degrading and present transiently. For therapeutic purposes, of course, I don't see that disease where you would need an injection of a weekly or 2 weekly or something so quite or monthly, that's might be not feasible. For that one, most likely, if it is a genetic disease, that similarly that using Cas9 mRNA, they permanently changed the genome and stop, the production of the toxic protein. And the other limitation is that the particle is going to surface closure cells. So even if you put in the CSF, for example, it won't go too deep into the neuron. It won't go through mucus. So there are limitations. You won't solve all of the problem with it. And you have to also consider that not the RNA is really the drug, but the produced protein. So my first project I was working is actually dystrophic epidermolysis bullosa, where the mutated collagen 7 protein, actually the half-life is 6 months. So you deliver the RNA, maybe the RNA is gone after 5 days, but the produced protein could be there for 6 months or more. So, and the same is for like the EPO, because it is 5 days is present, we could show in an animal. But of course, the red blood cells, which is produced, is there for 100 days. So, even extending, because the protein is the product. And I worked a lot in nitric oxide synthase, where the product was actually the nitric oxide, which is a couple of milliseconds. And then delivering the mRNA coding for the enzyme to locally, and we try to dilate the blood vessel with a product. So not the RNA is the drug, a protein, or maybe if the protein is an enzyme which produces something, maybe that is what is exerting the biological effect. But as I mentioned, I see all on this field is that the improvement of the formulation to make it that in vivo you can target, and that's a lot of effort is going to this direction, so that you don't have to isolate the cells out from the patient and put it back. But if you deliver the mRNA, which targeted to certain tissues, cell types, and this is already ongoing clinical trials for that. So this is improvement is coming. I don't see the RNA inherently has this extra hydroxyl, and we know that the dinosaur, when they isolate the DNA they can find, but nobody isolated RNA from dinosaur because it's quickly, and that's what it is.
Thank you very much, Katalin, and congratulations again from the Princesa de Asturias Prize.
Okay, the next question is coming from George Wick. George.
Okay, I'm an old professor for pathophysiology and immunology, emeritus at the University of Innsbruck Medical School, and I enjoyed, of course, the session. I just wonder how stable is the mRNA vaccine, because there are a lot of criticisms, I think unfounded, but nevertheless, that it's very stable, stays around very long, and may be the reason for the very rare but still present late effects such as autoimmune diseases.
Yeah, so that we had the criticism that it has to kept in -70 Celsius. That was the beginning, and yeah. I have to tell you that we kept
stable
it there
after injection. Sorry, perhaps I wasn't clear. Not stable as a vaccine in the tube, but after injection. How stable is the mRNA vaccine in the organism?
Oh, in the organism. So it is similarly what I have shown in an animal study. So it is the RNA degrades very quickly, and the produced protein also is not lingering longer than 5 days, definitely, and it exponentially degrades. The protein is the spike protein is not stable. And it is on the surface of the cells, but most of the particle is picked up by dendritic cells, because that is their function, to pick it up, of course. And so
it's volatile. So it's those papers that pretend that you find the mRNA all over the body, etc. It's just fake, isn't it?
So first of all, I have to mention that many to the toxicology study, and which was they found in the rat the different organ you might heard about, the different organ they could find, but the point was that it acquired. Okay, so what comes to your arm, it goes. goes to the animal. Okay.
I can't hear you, unfortunately.
Yes. Yes.
Okay. The next question, because we have several other questions, so will come from Anne-Marie Rafferty.
Thank you, Katalin. That was such a wonderful privilege to hear you speak, and a very exciting story it is too. I'm a nurse, but I'm also a historian, and so I'm interested in partly looking back as well as looking forward. And I just wondered whether, at what point did you get most excited by the potentiality of mRNA, and what were your imaginings, your fantasies at that point about what it might do? And could you ever have imagined that we might end up in the position that we have with the vaccine today? I hope I've understood your story correctly, because I'm coming at it probably more from a lay person. I'm not an expert in this area. But yeah, it's more about the story, the human element of the story. Thanks.
Yeah. Anne-Marie, I never ever imagined what is happened. You know, I never imagined that there would be, in our lifetime, there will be a pandemic. And I didn't even wanted to develop a vaccine. I made all of the modification and trying because I want to make it non-inflammatory. I imagine I work 17 years in neurosurgery. So with a colleague, physician, we try to use messenger RNA coding for nitric oxide synthase just to dilate the blood vessel, which is in a subarachnoid hemorrhage, one kind of stroke. It is a big problem. And they could reach that. And so constant talking to the physician to figure out what they can do, what I can do, and how we can work together. So that was the dynamic. And I imagine when in BioNTech, I wrote this grant about the dystrophic epidermolysis bullosa, that would be stimulating healing of the wound, for example, collagen 7 mRNA. And I imagine that people at their home, in their freezer, refrigerator, they will have some kind of mRNA. Everybody will have happened that, and they put it there and heal faster. Simple things like that. But not that 1 billion people will be injected with an mRNA. I was not expecting that. And of course, everybody would want it to go to as usual. Look at that, but. People were dying, and we needed, to accelerate and then not cutting corners, because actually every safety was checked and performed. And you have to know that the nucleoside modified mRNA vaccine were used in Germany. Mrna was already 200 people were injected with mRNA against bird flu. But it was not 200 million, it was 200. And of course, we wanted to go step by step. But, when the people were dying, it was just no time just to do the usual way. And of course, I was excited when I mentioned here the urokinase receptor, when I have first seen that it is functional. And then I imagined that we will use it for like bone marrow transplantation. Maybe older people can give bone marrow, and we can put telomerase and extend the tip of the chromosome. People can get that, so I was always basic scientist, but even when I worked with Eva Kondroshi, and we did, I always were thinking that, what will be good for something. And so human application was there in my mind. But actually my first project was an antiviral compound, and I worked on it. I didn't even know in 1978 that how much we would need an antiviral compound. And my PhD thesis, I worked in a modified nucleoside, because when I did this small trinucleotide, and so I was familiar with things like that. But yeah, so the human, I was always excited, and I was always happy. And as try to emphasize today, also the children in the school, that they have to find something they enjoy, because then they will good at that and they have happy life.
So I
Thank you. I
think all
look
of us
forward
will
to
have
the
so much-
television series. Thriller.
Thank you.
Okay, so the next question is coming from Francis Stewart.
Hi, I'm a new member and I work at the University of Dresden in Germany. I was interested in your comment about the optimization of the UTRs to raise the protein expression levels. What do you mean by that? What was needed to optimize protein expression by altering the UTRs?
So obviously, as Marin Kozak showed, if you have any structure in the 5' UTR, then you won't get the translation because the initiation complex doesn't have helicase, and so you try to avoid. And then sometimes complementarity also could be the 5' together with the coding sequence. So you might, depending on the coding sequence, you have to optimize specifically for the 5', and it is also critical. Some using very short 5', but it seems that a little bit longer one is better because the ribosomes are lining up, and structure free. It has to be a structure free, and the 3' UTR is. Again, we have the long poly A tail incorporated there. So we are transcribing because in one transcription reaction, we get all of these sequences. And this CAP1 structure, we are using the ClinCap from trialing, and the first nucleotide is A, not G. So we have 99% capping efficiency with this one. Yeah. So that's also, we had to change the very 5' end. And so this optimization, the backbone is there, and considering the coding sequence optimization, as you recently was just published, the SARS-CoV-2 case, out of frame translation product actually is also inducing very good immune response. So in my perspective, viral coding sequence should not be optimized because out of frame product could be also very beneficial protection. And then when you are putting more GC rich, then you are eliminating your uridine. And if you're eliminating uridine, then you get very long out of frame products because all stop codon has uridine in it. And so then you eliminate the uridine, then you get a lot of long out of frame product under stress. But
thank you.
And a lot of things is trial error, that's.
Okay, we are coming to the last question by Zoran Popovsky.
Thank you. I'm also a new member of Academia Europaea from North Macedonia, from Skopje. I have one general question related to RNA. Thank you, first of all, for your valuable presentation. How much reliable is the ratio, quantitative ratio between RNA and DNA as an indicator for growth potential in some tissues? I have some experiences with the fish muscles, but I'm wondering how much is reliable to quantitatively determine the ratio between RNA and DNA in some tissues. Thank you.
I mean, most of the work I'm doing in vitro transcription, but, best to my knowledge, in any cells, the RNA is minimum 10 times more than the DNA in total amount. But I am not the expert on measuring in different tissue the DNA and RNA ratio. Oops, my internet. Okay.
Okay, so we came to the end of the last session. Kati, I am extremely grateful to you for your availability. And I think your talk was really the highlight of this meeting. So thank you very much again. And if you wish, you can watch also the continuation. But we are going to have now 5-minute break, and then we are starting with introduction of the new members.
Yeah, and thank you, Eva, because I didn't mention on the slide, but actually that first experiment DNA with lipofectin we did together with Eva, and
If we wouldn't do that experiment, I would not be here today in Oviedo and other places, because that was the first inspiring experiment for me when we did that delivery with liposome. And we made the liposome, and didn't mention that we isolated the phospholipid for this liposome from the cow brain. We spent a whole week just isolating the phospholipid to make the liposome. So, and thank you, Eva, for all your...
It was really great, and thanks a lot. And I am very, very proud of you, and heartfelt congratulations to your new prize again.
Thank you.
Okay.
Bye.
Bye.
So welcome back, everybody. It's a great pleasure for me to chair the forthcoming session, which will be actually the introduction of the new members. The first session was today really fantastic with Katalin's presentation. Now I think also we can have some presentations today. During this session, I will introduce the new members. Some of you have already sent slides, and I will ask you, of course, to present them. However, those who haven't submitted their slides but would like to say a couple of welcome, of course, it's also possible. Anyone would like to ask for voice, please raise your hands in the virtual Zoom area, and I will immediately try to look at and give the voice to you accordingly. So let's look at the new members of Class C who were elected in 2020. And of course, it's almost needless to say that the chair of this session is Eva Kondroshi, who is a member of the Biological Research Centers at the Hungarian Academy of Sciences, Hungary. And the first session will be the biochemistry and molecular biology. And let's have a look. The first member is Edith Buzás from the Semmelweis University, and I'm more than pleased to give the words to Edith.
Thank you very much. Good afternoon. I would like to thank the privilege to say a few words about myself and my research focus. I work at Semmelweis University in Budapest, Hungary, and my primary research focus is extracellular vesicles, all kinds of different types of extracellular vesicles, including exosomes, microvesicles, microparticles, apoptotic bodies, autophagosomes, etc. And we are looking at the biogenesis, structure, and function of these vesicles in different physiological and pathological settings. Thank you again for your attention, and I hand over to our chair again. Thank you.
Thank you very much, Edith. It's nice to have you on board, and I wish you all the best and activity as a member of the Academy of Europaea. Thank you, Edith.
Thank you.
So the next member is Ilaria Capua from the One Health Center of Excellence from University of Florida from USA. I think she is not here. The next member is Ray Dixon from John Innes Center from the UK. And if I understood correctly, he sent his slides, and I would like to ask Ray to give some overview about his research.
Thank you very much. Can we have the first slide, please? Can we go back to the first slide? Okay, thank you. Oh, it seems to be slipping. I'm Ray Dixon. Thank you very much for the introduction. I work at the John Innes Centre in the UK, and we focus on biological nitrogen fixation, a process which is uniquely carried out by bacteria and archaea that convert atmospheric nitrogen into ammonia, a form of fixed nitrogen that can be assimilated by animals and plants. So what we aim to understand is how nitrogen fixation is regulated at the molecular level in response to environmental factors such as oxygen and fixed nitrogen, and in particular, how these multiple signals are communicated to the transcriptional apparatus via signal transduction pathways. We're exploiting this knowledge using synthetic biology approaches that aim to improve nitrogen inputs in agriculture. So now can we have the next slide, please? While we're waiting for that, I'll continue. Right now, we are facing a worldwide nitrogen crisis. Since although chemical fertilizers have been successfully used in supporting the growth of the human population, they're a source of huge levels of pollution and make a substantial contribution to climate change. On the opposite end of the spectrum, smallholder farmers in sub-Saharan Africa cannot afford to apply chemical fertilizers. So in contrast to chemical fertilizers, biological nitrogen fixation can provide a solution to the nitrogen crisis and potentially support more sustainable agriculture. Now, we already know that that's very successful in the legumes because the symbiosis with rhizobium is very effective and can supply all the nitrogen demands of the crop. But the problem is with cereals, because the contribution of biological nitrogen fixation by soil microbes to cereals is relatively limited. So we are seeking to enhance biological nitrogen fixation in cereal crops, taking the two different approaches which are illustrated on this slide. In the first approach, which is illustrated on the left, we're engineering ammonia excretion by plant-associated nitrogen-fixing bacteria to create synthetic symbiosis in which the bacteria donate ammonia to the crop in exchange for carbon provided by the plant. And in the second approach, illustrated on the right, we aim to engineer nitrogen fixation genes directly into plants to enable the assembly of the nitrogenase enzyme in plant organelles in which the nitrogen fixation machinery is protected from oxygen damage. So that's my introduction. I'm sorry it's a little bit long, but I'd like to hand you now back to the chair.
So thank you very much indeed. This is absolutely fantastic. And as far as we heard, it's really very important for our future. So what is currently the biggest challenge in your research, the questions you would like to answer in the future?
Well, there are a number of challenges which are illustrated here on this slide. One of these is to engineer a synthetic symbiosis. You have to be very, very careful about this because you have to get everything in balance. And the biggest problem with this is competition from other soil microbes. So we have to create an intricate symbiosis here between the bacteria and the plants. On the right-hand side, there's a tremendous challenge in actually introducing these genes into plants because there are very many different genes required for nitrogen fixation, and we need to get the proteins all in the same stoichiometry in order to work properly in an organelle. And we also have this problem with oxygen sensitivity, which is a particular problem in chloroplasts as opposed to mitochondria.
Okay, so thank you very much, Ray. It's nice to have you on board, and I wish you all the very best and activities at Academia Europaea.
Thank you. I'm delighted to be a new member.
Thank you. So the next member is Kristina Dinovic-Kariko from University of Vienna from Austria. She hasn't submitted her slides and most probably not be able to with us today. The next one is Duminel from the Pasteur Institute from France. Welcome, he's also on board. The next member is Ralf Erdmann from Ruhr University Bochum from Germany, and unfortunately he also couldn't be with us today. The next one is a new member also, George Fu Gao from the Institute of Microbiology from China. We have Fatima Geber also from the Center for Genomic Regulation from Spain. I don't know whether she would like to say a couple of words.
Yes, Can you hear me? Yes. Well,
Sure.
first and foremost, I would like to thank you for having elected me in 2020. It's for me really an honor to be part of this community. And I work in all aspects of post-transcriptional gene expression that are regulated by RNA binding proteins. RNA binding proteins are my main interest. And we're interested not only in how these master regulators of post-transcriptional gene expression recognize the RNA, but also how these interactions are distorted in diseases such as cancer. So that's it. Thank you.
Okay, thank you very much, Fatima, and thank you also for being here today and also involving with the research. And for example, can you say about this mRNA, not only in vaccines, but some kind of like therapies against cancers, or do you think this type of therapies can work in all cancers or just some of them?
Well, they are being tried. They're trying to be optimized for a number of cancers. Of course, every cancer is a different disease, and the efficiency is different. It's not the same to approach a glioma than a melanoma that is more superficial. So, but the hope is that these RNA vaccines, which are now, there is a resurgence of this approach now after the success of the RNA vaccines for COVID. Well, that there will be much research on this in the future, and that we will be able, I'm optimistic that we will be able to actually at least reduce the pathology of many cancer types.
Thank you very much. We need your optimism for sure. Thank you. So we go further. The next one is Pascal Ganchik from the National Center for Scientific Research from France. And I would like to give the words to you.
Okay, thank you. I also thank you for the nomination, and I'm happy to join the team, especially also Eva for probably propose my name. And so very quickly, the next slide just shows my cursus. So I did my PhD in Strasbourg, where I worked in the plant field on totipotency of plant cells and stress responses. And then I moved for my postdoc in the group of Witold Filipovic in the Friedrich Mischler in Basel. I changed also the topic because here I worked on RNA biology, so I entered in this world of RNA biology and also of the organism because I worked on human cells at that time and other cell types. And here I worked on actually modification of RNA. Especially 3' end modification with an enzyme, for example, which is RNA 3' phosphate cyclase. And then I had the opportunity actually to present a position in France, and I came back to the plant field where I worked on selective protein degradation. And I just say a few words on that part on the next slide. If you have it, yeah, the next slide. So we work on different mechanisms where ubiquitination and selective protein degradation, either through the proteasome or through autophagy, is regulating really important step in signaling, especially phytohormone signalings. We worked on ethylene, where you have some transcription factor that gets constitutively degraded, and when the gas, ethylene, which is an hormone, is produced under stress, actually these factors get stabilized. So we work on the mechanism, how a factor can be how the factor is regulated at the post-translational level. We work on other hormones like gibberellins or also ABA, which is important for stress response of the plant to drought and so on. More recently, actually, we are much more interested in the degradation and the post-translational regulation of the RNA silencing machinery per se, and especially on Argonaute protein. This machinery is really important for actually antiviral immunity in plants. And so we work both on on the virus side, where the virus escaped this machinery by specifically degrading some components of the silencing machinery, but also on the plant side, where post-translational modification events occur in this interaction. And you can see that the past always catch you up. So I could put together protein degradation and also RNA biology in a way in the same topic. And yeah, thank you.
So thank you very much, Pascal. I may ask a very obvious question, but you were working with plants and also mammalian cells concerning like stress. So my question is, what is the biggest similarity and also the differences between the plants and, for example, human stress response?
It's not really stress response. For the human, actually, it was not a stress response. We worked on enzyme. It was really enzymology and you know, the first cloning of this gene in human and to test it at the enzymatic level. But if you ask me this question, of course, it's well summarized what you have here on the cartoon. You see this plant is you know, it cannot escape. The insect can escape from all of this stress, and we can just put ourselves on the shadow. And the plant cannot, because it's a sessile organism, it has to live with all this stress. And for example, the number of F-box protein or these components which are specific for the degradation machinery, they are much bigger in plants, for example, than you can find in human. Human, you have 60 F-box protein. In plant, you have 700. And so you have a mechanism in plants where you can very easily and very quickly respond transcriptionally and post-translationally to many, many different stresses, and sometimes in combined way. So I think this is a big difference, is the sessility of the plant. So yeah, in terms of stress.
Thank you very much, Pascal, and all the best as a new member of Academia Europaea. So we have heard Katalin's presentation. It's, I think, needless to say that Academia of Europaea really very proud to have Katalin also on board, and we wish also all the best for her in the future, and we wish keeping her activities as well. So the next member is Michal Kubista from Czech Republic, from the Institute of Biotechnology. He unfortunately cannot attend today. We have also Joachim Lingner from EPFL from Switzerland, who works with telomeres and telomerase. He is also not here today. We have also Peter Marget from the Central National de la Recherche Center from France. We have Oliver Neyrols also from France, from the same institution. We have Valerie O'Donnell from the Cardiff University from UK, and she should be here. She sent slides, and now I would like to hand over the mic to Valerie. Please introduce your activities.
Thank you very much. I'm delighted to have been elected a member of Academia Europaea this year. Just to introduce myself and my work, I'm a biochemist working at Cardiff University. I mainly use mass spectrometry as a tool to identify and characterize new lipids. Particularly, we're interested in ones that regulate blood clotting and innate immunity. And the ones we've been working on mainly, I think, for the longest time have been the ones from platelets. But there are similar lipids made by neutrophils, eosinophils, and other blood cells also. The structures on the left are 4 structures that we found first in platelets. They're not present until you activate the platelets, so they're generated as part of the clotting mechanism. Since then, that number has expanded up to over 100, actually, that we now know are generated by human platelets. And what these lipids seem to do, at least from our studies, I've worked in collaboration with a number of clinical colleagues, Peter Collins and others at Cardiff, to look at their role in innate immunity and cardiovascular disease. And what we see is that they are made by the cells, and then they're externalized to the outer side, or some of them to the outer side of the plasma membrane, where they then allow blood coagulation factors to stick to the surface of the cells and do their job, which is to metabolize or cleave fibrinogen to form fibrin and then form a fibrin clot. We're also interested in how these lipids are regulating thrombosis in vascular inflammation. So not just in hemostasis, which is when you cut yourself and you want to stop bleeding, but also in infection, cancer, vascular inflammation such as cardiovascular disease and aneurysms. Also in COVID, obviously, there's a very big problem of blood clotting, so we're interested in the role of them there as well. and just generally, involved in lipid discovery and characterization, I think is sort of the main focus of the group. Also, separately to this, I'm currently the lead of a biomedical resource funded by the Wellcome Trust, which is called Lipid Maps. Originally started in the US in 2003, but in 2016, it moved to the UK. And this is a big collaborative resource that exists to curate in newly discovered lipid structures, to catalog them, and also then to categorize them into the various lipid categories, so whether they're phospholipids or glycerides or whatever. The Lipid Maps classification is kind of like the world standard now for classifying lipids. So that's a completely separate part of my job, is the leadership and the management with a number of fantastic colleagues actually running Lipid Maps. So thank you very much again. That's the end of what I have to say. Thank you very much. I'm honored to accept being a member of Academia Europaea.
Thank you very much for your short introduction. I mean, lipids play absolutely crucial role in many diseases. For example, in my research field, in pancreatitis, lipolysis has a huge impact and unfortunately worsen the disease. We also saw that in COVID-19, lipolysis can play a crucial role. So what do you think? Is there any way how we can modify the limits, the lipids in human to get the innate immunity better?
Yeah, so I mean, there are a number of lipids that are well characterized as modulators of innate immunity that you'll know of, like prostaglandins, for example, PGE2. So aspirin and ibuprofen are already out there as drugs that are used to dampen down inflammation. So they're certainly already big targets for anti-inflammatory therapy. When it comes to COVID, it's quite interesting. Emerging research from a number of groups showing that fatty acid oxidation by the host cells to support lipid biosynthesis may well be part of course, how the virus forces the cells to generate lipids to make its own membrane. And there's some research going on in the area of targeting lipid metabolism potentially as a way to reduce viral replication. And that's quite interesting. In our lab, we're doing some COVID research, but it's actually characterizing the lipids in the virus membrane itself, because this virus has an envelope. And, if this envelope is carrying lipids from the host cell, that could impact on how infective it is or how damaging it is to host cells, etc. So we're quite interested in the envelope and the composition of that, as opposed to host lipid metabolism, which is another massive complex area that we're only really beginning to understand right now.
So thank you very much, Valerie, and we also wish all the best for you as a member of Academia Europaea. So the next new member is Philip Poole from Oxford, from UK. Most probably he also cannot be here with us today. We have Zoran Popovsky from North Macedonia. He is definitely here. We heard his question, and he also has presentations to introduce himself. So please, Zoran.
First of all, I would like to express my honor to be part of Academia Europaea family. And the first opinion.
Maybe if you can go a little bit closer to your mic, because we can hear you.
Okay, is it better now?
Yes.
Oh, thank you. So it's really an honor to be part of Academia Europaea family, and especially because I'm the first scientist from North Macedonia in your family. My initial background is biology. I'm master in biochemistry. And I moved more than 25 years ago in applied area, and I defended my PhD in the field of agrobiotechnology. As a student, I was part of the Young Researchers of Macedonia organization, which was organizing a lot of activities among the students. After that, I started my scientific career in the National Academy of Sciences in the Center for Genetic Engineering and Biotechnology. And in the last 30 years, from teaching assistant to full professor, I'm at the Faculty of Agriculture and Food Sciences in Skopje. I'm leading the department for biochemistry and engineering. So as you can see, I'm mostly oriented in applied fields. I'm trying to motivate my colleagues at the Faculty of Agricultural Engineers to dig deeply in molecular mechanisms, and that's why my department and laboratories are serving as a service at the Faculty of Agriculture. So, in the Academy of Sciences, I was working in human medicine, molecular determination of the mutations in hereditary diseases, cystic fibrosis, hemoglobinopathies, muscular dystrophy. Then I was working on production of Taq polymerase more than 25 years ago, and we are producing that for our own purposes. We isolate another thermostable polymerase from autochthonic strain of bacteria here in Macedonia. And then I started with application of molecular markers in selection of domestic animals. Then I was using DNA microsatellites in determination of genetic distance in crop science. Currently, I'm leading also the laboratory for GMO testing. We were estimating the specificity and sensitivity of the techniques for GMO control. We were doing detection of and characterization of plant viruses. And most recently, I'm doing science in school program for our country. I have some background. I acquired some experience abroad, and also I'm inside the Faculty of Agriculture. I'm teaching here at the Faculty of Technology, Pharmacy, Natural Sciences, and Veterinary Medicine, mostly genetic engineering. I was Fulbright visiting professor at Ohio State University. I spent one semester at Adjunct University. In the last 2 years, I'm working at Novosibirsk State Agrarian University. I'm also a member of some bodies. Currently, I'm a member of the executive board of POST in the last 4 years. Please, next slide. And as you can see, my interest is very wide, and that's why I'm always surrounded by young people trying to infect them with the things that are now very, very contemporary. And unfortunately, we have always lack of human resources, but we are trying to do our best. Thank you.
So thank you very much, Ran, indeed. And I saw that you, as you mentioned, that you also promote science for youth. So how difficult it is in North Macedonia? So which population do you think science education should start?
So we established so-called creative center Kariko, and in the frame of that center, there are 10 small units. There are 10 primary schools in the previous municipality in Skopje, and we are doing science during the teaching process. We are developing experiments during the teaching process, and it is our target now to work with the children 14, 15 years.
Okay, thank you very much indeed, and welcome on board. So the next member of the biochemistry and molecular biology section is Sheena Redford from University of Leeds, from UK. We also have a new member from Germany, Mrna Rodnina from the Max Planck Institute for Biophysical Chemistry. We also have Francis Stewart from Germany, from Dresden. And we also have, I am not sure if she, or Stuart is here. Francis?
Yeah, I'm here.
Okay. So maybe a couple of words from you.
Okay, I'm sorry I didn't send the slides, but my lab focuses on epigenetics. We discovered the first histone 3 lysine 4 methyltransferase and contributed to the realization that the methylation status of the histone 3 tail in nucleosomes is a primary determinant of epigenetic regulation. So we continue to work on that topic, largely in mouse development, so in mammalian development, and also utilizing embryonic stem cells as the model system. but my lab is probably best known for the technology we developed. We developed the technology because studying epigenetic issues is rather complex, and we were convinced that we needed better tools. And so we managed to come up with a few contributions to genetic engineering, particularly ligand-induced site-specific recombination, which in mammalian applications is tamoxifen induction of Cre estrogen receptor fusion proteins. So that's a lab original. That was one of my first papers as an independent group leader with my first PhD student. And also the technology you may have heard of called recombineering, which is the use of homologous recombination mediated by phage proteins for recombinant DNA engineering. It's particularly useful for engineering large things like bacterial artificial chromosomes, which are beyond the size of, conventional restriction enzyme, PCR, ligations technology. So, the two main themes in the lab, epigenetics and genetic engineering technology.
Okay, thank you very much, Francis. We know that epigenetics play a crucial role in many, many diseases, or maybe can we say that in all diseases they play a role, or maybe mostly in cancer development, or there are specific diseases which epigenetics play bigger roles?
It's definitely incredibly important in cancer mechanisms, so that the International Cancer Genome Sequencing projects, I think the biggest surprise was the frequency of mutations in epigenetic regulators, and particularly the ones that we work on the most, which are the MLLs, the mixed lineage leukemias. There's 4 of them, and they're amongst the most highly mutated genes in all types of human cancer, and we're still lacking a decent explanation for that. I think epigenetic mechanisms in diseases of aging are also quite prominent. So those two issues.
And they are mostly like associations or initiations with cancer. So maybe smoking acts in both fields, or what is the real connection between the two? So most probably there are plenty of evidences that epigenetic changes comes first.
It's a very good question. Really, there's only a few cases where it's really proven that the epigenetic regulator is the cancer driver. And in most cases, it's just an association, and we're still lacking the evidence. Whether it's a passenger or a driver, still open questions.
So it means there are lots of things to do in the future then.
Yes, yeah. So my postdoc supervisor advised me to pick a topic area that will never be solved in your lifetime.
Okay, thank you very much. So I wish you all the best as a new member.
Thanks for having me, allowing me to join.
Thank you very much. So our new member is Gian Tartaglia from Italy, from University of Rome, Sapienza. We have another member from Saarland University from Germany, from Jörn Walter. I don't want to ask every time who is here and who is not, because we would lose plenty of time. But if somebody is here, just don't hesitate to immediately raise your voice. We have Yun Jui Yang from Beijing Institute of Genomics from China as a new member, and I think this was for this section. And the new section is the cell and developmental biology, which is led by Crisanto Gutierrez, actually from Spain. So we have a new member, Philippe Bosso from the Pasteur Institute from France. We have Joseph Grunbaum from Jerusalem, from the Hebrew University, from Israel, and we have here, most probably from Spain, from Crisanto Gutierrez, and very welcome. Thank you for sending your slides, and now the stage is yours.
Okay, thank you very much actually for this introduction, very short introduction. And of course, I am delighted and very honored to be part of the academia. So I'm looking forward to work for the academia in the coming years. As you say, now I'm currently the chair of this cell and developmental biology section. And I'll be, of course, very pleased to receive any feedback from any member in order to improve the activities. Can you go to the previous slide, please? Yeah, I work in the Center of Molecular Biology in Madrid. And over the years, I have been interested in several aspects of a cell cycle in general, with different aspects, and with the main focus on how things happen during the cell cycle progression and during genome replication. So I've been interested in this aspect, mainly using Arabidopsis as a model organism. And in most recent years, the challenge is, as shown in the next slide, please, is to understand how things happen not only at the cellular level, but how these processes are integrated in a whole organism during organogenesis. And in the middle part of the slide, it is shown here that from the point of view of cell proliferation, there are stem cells that, of course, produce derivatives. They enter the cell division, produce many cells that eventually take different cell fates, differentiate, and form parts of the organs. However, our main interest is to understand not only the dynamics between these different cell populations, but also how the hormonal, developmental, and environmental signals are integrated into this cell population dynamics. We have developed a number of tools to analyze this in vivo. And of course, as just mentioned in the previous talk, one main component of this regulation is epigenetics and chromatin modifications. So then what we are trying to understand is how all these processes are integrated to learn how growth is produced in plants and how we can eventually manipulate this growth in the benefit of producing better crops or better yields. So it's not included here, but the work is currently funded by both national agencies and also from the European Research Council. So thank you very much.
Thank you very much, and of course I think it's an evergreen question about from stem cells during the organogenesis, as you showed your slides, and one of the biggest challenge is the differentiation about whether how the organ can develop, and of course the biggest danger is about the cancer development. So what is new in this field, and what is our hope for the future?
Of course, there are two aspects that we can discuss about that. One is the high ability of plant cells to de-differentiate and come back to the stem cells of their early stages in development. And this is terribly important in order to produce regeneration. But also, as you mentioned, something that we have been involved for years has been to analyze the function of both homologs of tumor suppressor and oncogenes that are identified in humans, and actually they are present in plants, of course, because actually they evolved earlier during evolution. In this case, it's extremely important to know the balance of this retinoblastoma and E2F and other cases in order to understand the dynamics of this population and their role in differentiation and cell renewal.
Okay, thank you very much, and all the best also for you and for your section at Academia Europaea.
Thank you.
So our next member is Dirk Inze from the Center for Plant Systems Biology from Belgium, and he's here with us, so the stage is yours.
Yeah, thank you very much, and thank you very much for this election to become member of this prestigious academy. Again, I think it's really a place, like an organization which can have an important influence on the science activities in Europe. So I'm from Belgium. I work at the University of Ghent. I'm also the science director of the Flemish Institute for Biotechnology, but I'm responsible for the plant research. And I work already for many years on the molecular mechanism of, and actually the molecular networks of organ growth, particularly interested in what determines actually the size of plant organs. And most of that work we do on leaves, but it turns out that many of these mechanisms are conserved in seed development, in crop productivity. And so we do that both under, let's say, plants grown under normal condition, but also very much how environmental conditions like drought negatively affect the growth, and what are the molecular pathways behind all that. So much of our research is actually done on Arabidopsis, but we're doing a lot of things also in crops, particularly in corn. And over the years, we have been very active in translating this knowledge actually on crop improvement up to the level of field trials, like nowadays with genome editing crops, both in the EU and in the US, in fact. So maybe it's important to mention here that this work has been supported by 2 advanced ERC grants, and the latest one is really combining genome editing at the multiplex scale, with really sometimes hundreds of genes, in fact, and combining this with breeding. Also, I think maybe important for this academy is that I'm also, since last year, member of the ERC scientific board, in fact, and which is, I think, has an important influence on how this organization works. And last but not least, I would like to mention here that I'm the founder and the coordinator of an international non-profit organization, which in fact is working on science-based policy to enable, in fact, the use of genome editing in Europe for crop improvement. As you know, the European Court of Justice recently ruled that genome editing is, in fact, or GMOs, which really makes it extremely difficult to apply this technology for crop improvement in Europe. And this is really in contrast what's happening in the rest of the world, which are really going full force. And this EUSH, in fact, is now 134 members, different institutes across Europe, scientific plant science centers with joint forces, really to bring this kind of policy-based, science-based policy really on genome editing to the highest political levels. And maybe that's also interesting, maybe to discuss this in detail at another occasion. Thank you.
Thank you very much indeed. And when I saw your slides, my immediate questions, which shows in my head that how difficult it is to translate your work and the benefits of gene editing to public language or the people's language. And what do you think, for example, Academia Europaea can help on it? Because I think these issues must be solved, especially in Europe.
Yeah, it's extremely important to have a kind of multidisciplinary approach involving also a psychologist, particularly social psychology is really important. And I think we learned, we are collaborating with a team in Poland, where we learned how to make narratives, in fact, which are really bringing this technology in a kind of understandable manner to the public at large. Certainly what we should not do is to fight, to react on all this negative anti-GMO legislation and people, because you just to reinforce the message. You have to bring it in a very positive way, and really highlighting the benefits, and at the same time really connecting with all stakeholders in the field, because too often scientists preach for scientists, and that's too limited. You have to reach out to the public at large, to politicians, really emphasizing the advantages of the technology and for making a more eco-friendly, sustainable agriculture. And that's, I think, important. And that involves scientists from really other classes, like, in fact, and there an academy, because you represent all this diversity, could really help a lot.
Thank you very much. And I think, for example, what I found in Academia Europaea, that they are very happy to have proposals about what kind of actions and Academia of Europaea can help towards EU and everything. So I think please feel free to submit your action plan proposals to the board of the Academia Europaea. I am 100% sure that they will help developing documents, materials to help to solve this kind of questions. Thank you very much being with us.
Thank you. Thank you.
So the next member is Lars Nitschke from the University of Erlangen, Nuremberg from Germany, and I think he is with us. He sent his slides, and Lars, the stage is yours.
Thank you very much. I'm also grateful to be elected in this academy. So I'm an immunologist from the University of Erlangen. I work on B-cell immunology, autoimmune diseases, and we apply genetic mouse models, and we are interested in B-cell signaling. So next slide. So the B cells are, of course, the cells of the immune system which make the antibody responses, and what you see is these sorts of inhibitory receptors we work on recently, CD22 on the left side and cyclic G on the right side. They're both inhibitory receptors. So if a B cell is activated by antigen binding to the antibody called B cell receptor, signals are triggered, and this is negatively regulated by these inhibitory receptors. Next, please. So when you delete these inhibitory receptors, you see higher signaling in the B cell, which you can see on the left bottom side. And next, also on the right side. So with the loss of these inhibitory receptors, there is a stronger activation of the B cells. Why is this relevant? This is relevant because this may affect autoimmune diseases and also leukemia formation, because if these inhibitory receptors are missing, there may be autoimmune diseases induced. Next slide. And we could show this in mice which lack these inhibitory receptors. For instance, cyclic G deficient mice develop autoantibodies, which are typical for diseases like systemic lupus erythematosus. Or in the bottom, cyclic H deficient mice after a virus infection develop a kidney nephritis, which is also relevant for this disease. Next slide. And the last slide shows that when cyclic G is missing, there's also a leukemia formation, which is much stronger and happens much earlier. You see in the bottom these leukemic cells, which are circled, which occur in this TCL1 model for CLL. And in the case of cyclic G deficiency, there's a stronger development, which you can also see from the purple bars in this upper panel. So these inhibitory receptors are relevant for these diseases, and we would like to translate this into the clinic in the future as well. Thank you very much.
Thank you very much indeed, and maybe I ask you something what plenty of times asked about the controversies, or maybe it's just only controversy in myself, that how useful is measuring in COVID-19 about the levels of antibodies and how much the level of antibodies actually can correlate with the host response.
Yeah, so the antibodies is one thing. What you need is the neutralizing antibody, neutralizing antibodies against the spike proteins. They are a good indicator. But there's, of course, more than just the neutralizing antibodies. They may drop after a while, but you then have also the memory cells, both memory B cells and memory T cells, which stay on for quite long, both after infection and also after vaccinations. They are more difficult to measure, but they are very important for the recall response. So antibodies is just one indicator of the how the disease is progressing and your protection is progressing, but the memory cells are also very important.
So would you measure for everybody, like after the second vaccination, the neutralizing antibody level, anti-spike one?
Yeah, they're a good indicator, of course. Yeah, I mean, you can, especially if you follow this over time, you will see that it will potentially drop after a couple of months, and that may be different from individual to individual. So this is a very good indicator how good your protection is. But I'm only saying, even if that drops under a certain level, there may still be protection, which is not so easy to measure and easily if we go to a practice.
Thank you very much updating us, and thank you for being a new member of Academia Europaea.
Thank you as well.
So the next member is Arturas Petronis from Vilnius University from Lithuania, and I think he's also here with us.
Yes, I am.
And the stage is yours.
Thank you. Thank you for the kind introduction. Can you hear me okay?
Yes, very well.
Great. Okay, so can we move to the next slide, to the single slide that I have? Great. And can you start animation, please? Yeah, so the take-home message of my 3-minute talk is that cyclical epigenetic processes may play an important role in human disease. And it's not moving yet, so can you press the button? Yes. So, and I think we can keep going this recycling again and again and again while I'm talking, and you'll understand the reasons of the message from the slide. So the goal of my research effort is to understand the causes of human disease, primarily psychiatric ones, such as schizophrenia, bipolar disorder, and major depression. About 20 years ago, I suggested that in psychiatric and other non-malignant diseases, epigenetic factors could play an etiological role. Epigenetic factors are small molecules attached to DNA and nuclear proteins, and you already heard this term before during the earlier presentations. And in very kind of general sense, epigenetic factors perform various regulatory functions in the genome. So the best example of the importance of epigenetic factors is each of us. Each of us is made of thousands of different cell types, and the genomes, if we talk about the same individual, the genomes of such cells are identical or nearly identical, but cellular phenotypes and cellular functions can differ dramatically. So just think about neurons and muscle cells and skin cells. So this kind of diversity is achieved, at least in part, due to differential epigenetic regulation. So jumping over a number of specific arguments, we have hypothesized that even small deviations from normal epigenomic profile in the cells and tissues can be an important disease risk factor. So the general understanding over the last several decades has been that in disease, epigenetic factors gradually deviate from the norm in a linear fashion. Now you can repeat the animation again so that the audience would understand, would notice the dynamics. So, so this is the general understanding that things are changing in linear fashion, and epigenomes are changing in linear fashion. There is either loss or gain of epigenetic marks. But it's not clear why some genes deviate from the norm and others remain stable. This is a big question. So several years ago, we discovered that some epigenetic sites, epigenetic factors, can change in a cyclical fashion. More specifically, we focused on circadian oscillations, which are 24-hour cycle. So such cyclical epigenetic oscillations seem to be linked to linear ones, and linked in a very interesting ways. It looks like cyclical oscillations precede linear changes, and cyclical oscillations may even cause some linear epigenetic changes. So now we're trying to understand if deviations from the normal epigenetic rhythmicity can be an early hallmark of disease. And there is some evidence that actually different individuals in different patients oscillate in different ways. There are some similarities, of course, but there are also some differences, which leads to some interesting ramifications to personalized medicine. So what is shown in this slide is that actual oscillations could be two types of oscillations: normal physiological oscillations, which are marked by green bubbles, and disease or pre-disease related oscillations, which are shown as red bubbles. And all this entire thing is oscillating. And there could be some places, some ectopic places of oscillations, or some changes in oscillation parameters like amplitude or phase, which plays a role in disease. So this is the key, the main hypothesis, the main theory that we are currently working on. Thank you for your attention.
So thank you very much. It's fascinating. And I just would like to ask that how specific these epigenetic changes in the disease. Of course, it's a longer period which needs to get these changes. So whether there is a stage when we can predict with high probability the disease onset, or more importantly, if we can predict the disease onset, is there any way to interfere and maybe delay or cancel the onset?
Yeah, this is a very good question, actually. There is no answer yet, but that's probably the most important question that we are trying to address. If we could identify deviations from normal oscillations before the disease onset, that would be like a hallmark of disease that could be used for disease prediction, and possibly using different strategies, starting with the kind of traditional pharmacological agents and ending with epigenetic editing, we can try to rectify the pathological oscillations. But again, like the key question is, the key kind of message here is like how early we can detect deviations from normal oscillations, which eventually would result in disease, bona fide disease symptoms and onset and all those things that already basically is quite often it's already too late to do something efficiently.
Yes, thank you very much, and all the best and good luck for this very important vision, mission, and specific goals that you have also as a member of Academia Europaea.
Thank you very much.
So we have again a new member and cell and developmental biology group, Mauro Piacentini from Rome, from Italy, and he shared his slides with us, and now the stage is yours, Mauro. Or maybe he's not here. If maybe some connection problems, please interrupt us again and we will come back to you. So the next one is Antonius Rabeling from Leiden University Medical Center from the Netherlands. Our next member is Alexander Sier from the University of Basel, again from Switzerland, and I think it was the one for this session. And the new one is the Physiology and Neuroscience, led by Balázs Gulyás from the Karolinska Institute. And let's look at the first member is Stuart Allen from the University of Manchester. From the UK, we have Francesco Paolo Battaglia from the Netherlands, from Nijmegen. On stage, we have Fabio Benfenati, also from Italy. On board, we have Antonio Cattaneo from also from Italy. We have David, and maybe he is here with us. We have Fabio here.
Hi. So first of all, I want to say that I'm really honored to have been accepted in the Academia Europaea. I will be very brief. I'm a neurologist and neuroscientist, and I always worked on the molecular mechanism of synaptic transmission and plasticity, and more recently, how these processes are altered in neurological diseases, particularly related to epilepsy, autism spectrum disorder, and neurodegeneration. So besides having clarified many of the molecular mechanisms and actors of synaptic transmission, more recently I've been involved in creating hybrid interfaces between smart materials and neurons, including optogenetics and contact with photosensitive materials to be able to modulate neuronal activity, both electrically and metabolically, with light. So this is sort of an advanced field of investigation that has led us also to propose a new approach for retinal photoreceptor degeneration using these photosensitive materials, which can convert light into an activation of the inner retina, and thereby being proposed as new strategies for retinal prosthesis, for artificial retina.
Thank you very much indeed. It's a very interesting area. I am not in the field. I know Kondroshka's experiments concerning the optogenetics, and I don't know whether are you collaborating or any kind of interaction between your labs?
We have a European project together with Botond, and yeah, we know each other well. Yeah.
All the best for you, and thank you for being with us today.
Thank you very much.
So we have David De Gregorio from the Pasteur Institute from France. We have here George Gagalashvili from University of Copenhagen from Denmark. We have Matthias Höhn from Germany, who is working in experimental stroke research. We have Anthony Holtmat from the University of Geneva from Switzerland. We have Siegfried Kasper from Vienna, from Austria as well, working on many different diseases and try to find therapeutic targets. We have again Barbara Koffler from Austria, and I think she's with us, and we are absolutely interested in what is your research and what is your major challenges in the future.
Okay, thank you very much. I also thank the board for selecting me into the Academia and Europaea. I'm by training a chemist and later on a biochemist, and after a PhD postdoctoral stay in Sydney, Australia, I joined the University Hospital in Austria. There, I'm working on two different topics. One is related to the function of neuropeptides and regulatory peptides, especially the peptide galanin and its family members. We are investigating the function of these peptides in immune cells, inflammation, and metabolism, and also the pharmacology of their G-protein coupled receptors. The second topic is completely not related to that, but that is related to the investigation of mitochondrial energy metabolism and genetics, where because of these changes in tumor metabolism, we are trying to target tumor metabolism, mainly by dietary intervention with a high-fat, low-carbohydrates diet. And we are combining these ketogenic diets with classical types of therapy, especially chemotherapy. And now we are going to investigate also the combination with checkpoint inhibitors and antibody treatments of cancer. And we are working on a lot of different animal models to answer all these research questions. Thank you.
Thank you very much indeed. The first field, the neuropeptides, I remember from my PhDs in the 90s that it was, of course, neuropeptides and research on the top. So concerning the neuropeptides, what is the biggest challenge nowadays in it. And my second question would be about the ketogenic diet, but because, for example, in pancreatitis, we also look at about what kind of diets would be the best for our patients. And of course, there is evidence concerning the short-term beneficial effect, but do you think that ketogenic diet in the long term could be beneficial, or there are lots of dangers which are not seen yet?
Okay, to the first question related to the neuropeptides and the challenges, I think the challenges are still the same, that we need specific tools and non-peptidogenic specific ligands crossing the blood-brain barrier if we are looking at brain diseases. And the other one is that they are so diverse in their action that it's really hard to pin down their major functions. And so interference with them is not that easy. Regarding the ketogenic diet, I think for maybe over 80% of the cancer types, at least it will not do any harm and eventually delay tumor progression. What we have seen recently is that the ketogenic diet and low-carb diets increase quality of life of cancer patients, which is also very important. The experience with ketogenic diets in epilepsy patients who are pharmacoresistant and are on a ketogenic diet over years and years do not indicate any severe health problems if you are on a ketogenic diet for a long time. We think what could happen is in tumor therapy, though, is that once you are on a ketogenic diet, you might not be able to go off the diet because otherwise the tumors might start to proliferate again. But that's not completely clear, and there are a lot of clinical studies missing to make that point clear.
And this ketogenic diet can help to avoid sarcopenia for cancer patients as well, or can delay it?
Yeah, obviously yes, but still there are not enough clinical trials, and in mice are not that similar in that respect. So we are definitely lacking those studies. There are only very small, not very well controlled studies. And I think the field is moving forward, and the tumor metabolism field definitely, nearly every big group is trying to apply ketogenic diet in one form or the other.
Sure. Thank you very much indeed, and thank you for being a member of Academy of Europaea.
Welcome.
Thank you. Another member is Jean-Pierre Madet from Paris, from France. We have another new member in this group, Sandeep Patel from the University College London. From the UK, we have another member from Sweden, Marcela Pekna, who is working in neuroimmunology as an interdisciplinary way. And here, what I just mentioned, we have Kondroshi, who is, of course, working to rebuild retina, and I think he's here with us. Or maybe he also couldn't join in. We have Annette Schenk from Radboud University Medical Center from the Netherlands, and she sent her slides, and she is here with us.
Yes, hello. So my name is Annette Schenk, and I'm leading a research team at the Radboud University Medical Center located in Nijmegen, which is the Netherlands' oldest city. And my group uses genetic, neuroscience, and molecular approaches to generate and characterize disease models. And the powerhouse of our research is the mighty fruit fly Drosophila. The diseases of our interests are rare monogenic neurodevelopmental syndromes, above all associated with intellectual disability and autism. In the past decades, hundreds of disease genes have been identified, providing us really exciting starting points into understanding brain function in health and disease. And making optimal use of this precious information requires a highly efficient model, and this is exactly why we work with Drosophila. Can I have the next slide, please? So taking advantage of this powerful toolbox that Drosophila comes with, we perform in-depth mechanistic studies into specific disorders. We conduct large-scale phenotyping of hundreds of disease models to uncover the most disease-relevant fly phenotypes and their underlying molecular networks. We characterize novel candidate disease genes to support our diagnostics and develop novel treatment approaches. I have time for a short example to fill out the outer two pillars with a bit of content for you. So can we move to the next slide or to the animation? Thank you. So our most recent study tackled a frequent problem in autism and intellectual disability disorders, which is sleep disturbances. They can dramatically affect cognitive functioning of patients and quality of life of their families, yet they remain severely understudied. So we focused on two disorders caused by mutations in two closely related genes. And we found that patients, as well as our Drosophila models, show specific problems in sleep maintenance, causing highly fragmented sleep. And we narrowed down the origin first to glial cells, and then please, the next animation, and subsequently to a specific glia subtype that forms the blood-brain barrier. We found that loss of the Drosophila autologue in blood-brain barrier glia, please, the last animation, causes dramatically increased levels of serotonin underlying the sleep fragmentation. Remarkably, despite the developmental origin of these defects, we were able to completely reverse them with a behavioral regime resembling a human sleep restriction therapy. I just say like this, fine. So this study provided novel insights into glial regulation of sleep, uncovered the first genetic cause of hyperserotonemia, which is the most replicated autism endophenotype, and linked it to blood-brain barrier dysregulation and autism-associated sleep disturbances. Plus, it uncovered a directly translatable therapy that we are currently implementing in patient care with our clinical collaborators. So this is a typical symbiosis of fundamental and translational expertise that I'm happy to add to this community. And thank you very much for permitting me.
Thank you for being with us. And I think all scientists can ask you how much sleep would be normal and physiological in our life, because I think everyone is struggling with this question.
Yeah. I do agree. Sleep is only one aspect we work on, but obviously the answer is that this is highly personal. And actually also in our sleep restriction therapy, when we work with mutant Drosophila models, we always need to tailor it really to the genotype. Yeah, so to just, the idea of sleep restriction therapy is that you actually match sleep needs and sleep opportunity. So we have also these flies, when we want to treat them, we have to give them exactly the right dose of sleep opportunity, so that actually in this opportunity they sleep most efficiently, because it's not only about the total length of sleep, but it's also the quality, and this is actually what you can control with the sleep restriction therapy. It's a highly exciting tool, and actually also the link between sleep and cognition, not only that sleep is necessary for cognition, but that it can be positively implemented for enhancing cognition, is actually, I think, very exciting topic.
Thank you very much indeed, and thank you for being with us. And I think I missed somebody, if I looked at the chat correctly, because I was very quick earlier. I don't know whether Thomas is still with us.
I'm still here, thank you.
Okay, I am so sorry, miss you about, and please introduce yourself.
Okay, so I don't know if you presented a slide with me with my name, but my name is Thomas Halliday. I'm from Sweden, and my area of research is DNA repair. So what we do in DNA repair, I don't know if you can, I'm in cell and developmental biology. So what we are doing, I don't think that you have, do you have my name there? No. No, I think you missed my name altogether. Yeah.
Okay, we are very, very sorry.
Okay, that's okay. My area of research is DNA repair. We work on the basis that cancer cells have more DNA damage than normal cells. And the key is that what we do then is that we make inhibitors to DNA repair and exploit the mutations in cancer that increase these mutations. So we can see, for instance, with PARP inhibitors, that PARP inhibitors can be used to kill cancer cells. So this is something that we have done in my lab, and this has now been translated into the clinic and is now an approved therapy for BRCA-mutated ovarian, prostate, breast, and pancreatic cancer. We have continued with this work, and we have identified the basic repair pathways that DNA replication forks, and also then in my lab created a multidisciplinary team where we have medicinal chemists and we have pharmacologists. So we have a lot of medicinal chemists and pharmacologists, and with that we can develop new inhibitors to new proteins. So we do first-in-class inhibitors to MTH1 inhibitors, which we have taken ourselves into phase 1, now entering phase 2 trials in cancers. We have also discovered OGG1 inhibitors, that is inhibitors to repair of oxidative DNA repair. So in particularly, this is interesting because what these do is not only repairing DNA, but they can also sense oxidative damage. And this is very common in many diseases, such as inflammation. So we can see that OGG1 actually bind to pro-inflammatory promoters to bring about pro-inflammatory transcription. And with inhibitors, we can then block the pro-inflammatory transcription. So this is a new way of treating anti-inflammatory responses. So this is something that we are also taking towards the clinic. And finally, we are also have identified new OGG1 activators. That is taking back a more ancestral role of OGG1, this protein repairing oxidative damage. So what it does is that it's regaining a more efficient role. So it becomes 10 times more effective, repairing oxidative damage. And it does this, prevents senescence and aging. And it also then is not residing at the DNA. So it's no longer can trigger the inflammation response. So here we think that we have a new treatment, not only for those diseases or those affected by oxidative damage, but also to prevent inflammation.
Thank you very much. This is really, really very interesting and new area of research. I just asked because as far as I understood correctly, but maybe my knowledge is not excellent in this field, that for example in advanced prostate cancer they are not in the first or second line therapy, but they are in the third or fourth line therapy in BRCA positive patients, these therapies. So or maybe they should be in the first line therapy in the future.
So they are added up to so right now they are approved in castration resistant prostate cancer. Sure. As a maintenance therapy. When it comes to ovarian and breast cancer, they are approved in first-line therapy, so as a maintenance therapy, and that improves progression-free survival from about 1 year to more than 5 years. So they have a great advantage for those patients, and we know that when you get in with a PARP inhibitor later on, when you have chemo-resistant cancers, that they are less effective, because we inhibit DNA repair, causing DNA repair damage, such as caused by chemotherapy. So if they are resistant to chemotherapy, they're also kind of resistant to our treatments, more or less.
Thank you very much indeed, and welcome on board.
Thank you.
Thank you. And I think we may miss also somebody who wanted to say some words. I don't know whether she or he is here.
I can say a few words.
Yes, please.
Hi, I'm Sandip. I'm a professor at UCL here in London. Hi, Tim. I'm a molecular cell biologist. I'm interested in calcium signaling, and my work over the last 20 years has helped develop the concept that acidic organelles, such as lysosomes, are actually signaling centers involved in calcium homeostasis. So we've been trying to define channels and transporters which allow calcium to go in and out of these organelles. We've been trying to work out the cellular processes which these fluxes regulate, mainly to do with membrane traffic. And increasingly, we've been trying to associate these fluxes with diseases, in particular Parkinson's and viral infection. So thanks to the Academy for having me.
Thank you very much. I think it's a very important question. As a pancreatologist, I know this acid load in acinar cells, where there are plenty of very acidic lysosomes with plenty of actually inactivated enzymes, and the calcium signal can totally make it activated and start the whole processing. Do you have any research in acinar cells as well?
Yes, it's interesting. The acinar cell actually has been really instrumental in trying to work out the basic signaling pathway through these acidic organelles. It was the first mammalian cell type in which it was recognized that these acidic organelles can actually function in calcium signaling. So yeah, we've had an interest in cholecystokinin signaling in the pancreas, its production of second messengers, which then go on to release calcium from these stores, and also in the neighboring beta cells, which of course in vivo will work together.
So there are still lots of things to do. So thank you very much for sharing your words with us. And now we move back to our presentations, and we have here Eva Kondroshi from University of Rome, from Italy, yes, and he also sent his slides.
Yes, I'm here. Yeah, I want to thank the Academy. I'm really honored to be part of the Academy. Actually, I'm an immunologist by training. My first part of my career, I work on apoptosis in T cells and how apoptosis process regulates the immune system, including the neuron immune regulation process. Next slide, please. And recently, I moved my research to mesenchymal stem cells with a specific focus on how mesenchymal stem cells interact with the inflammation. Actually, the mesenchymal stem cells exist in almost every tissue, and the cells can be easily isolated and expanded in vitro, so you can study them in vitro and in vivo. And this actually plays an important role in regeneration and tissue repair. So those cells have a unique property after interact with the inflammatory cytokines, and they get activated and dramatically increase their metabolism, especially glycolysis. So basically they can suck in all the glucose and then make tons of growth factors and immune regulators. So those regulations actually can be thousands of fold. So this really gives a new way to look at regeneration. So in the dogma is for regeneration, you have a damaged cell, then you give the stem cell to replace the damaged cell. But our study showed that the majority of this regeneration process, in fact, is due to those cells get active by inflammation and produce various growth factors. And those factors actually wake up the endogenous stem cells to have the tissue repair. So we call this process called cell empowerment rather than cell replacement. So of course, in tissue, you have a various type of inflammation and a various degree of inflammation. So those play very complex roles. So we are looking at how those cells interact with the inflammation and how the growth factors, cytokines, form a new network during wound repair, during disease process, and most importantly, during the cancer development, because cancer really is also a wound that never be repaired. So I'm looking forward to work with the colleagues in the academy and I hope that this can bring some new treatment to the patient. I'll stop here.
Thank you very much indeed. I think these kind of stem cells are really very important in the gastroenterology field. As far as I understood, I learned that, for example, in gastric local cells, stem cells, when there is an injury coming, they can help in repair. However, it strongly depends on the differentiation of these kind of cells, because as far as I understood, if they coming from central part and they are not as differentiated as they should be, they make damage and cancer development. So how can you interfere to this process and maybe have these stem cells to differentiate?
Yes, I think this differentiation is very, very important. In fact, the inflammatory cytokines can interfere with the differentiation process. And recently we have a look at how the differentiation into the, for example, into adipocytes get interfered by inflammatory cytokines. So we have a paper just prepared. And also, under the inflammatory cytokine situation, those cells can also produce factors that can regulate, for example, metastasis. We have a paper going to be out related to the production of C3 from the new niche for cancer metastasis. So this differentiation process actually can be interfered by inflammation.
Thank you very much indeed, and thank you for being a member of Academia Europaea. We need your optimism and enthusiasms. Thank you. So I just got a message from Maria, who has to leave to another meeting. I don't know whether she's still with us, and maybe can share about her notice or her knowledge research field with us. Or if she's already left, we can move forward. And the next member is Gerhard Thiel from Darmstadt, from Germany. I don't know whether Gerhard is with us. Here he sent his slides.
On board, yes. Hi.
Hi.
I'm very proud to be a member of the Academy. And I have to say, this was really the highlight of the last year in quarantine, sitting at home and being depressed, and to get the notice that I was elected was really one of the big highlights. A few words on my science. My science is, I would say, really in the spirit of the European Academy, because I share all my work with my wife, and we have a transalpine laboratory. Part of it is here in Darmstadt in Germany, and the other part is in Milano in Italy. So it's really going across back and forth the Alps. And if you have the next slide, please. Our hearts are beating for ion channels. And by serendipity, we found, together with colleagues in America about 20 years ago, that these very big viruses, DNA viruses, which you see on the left, they encode in their genome for proteins, which are like the potassium channels in our brain and our muscles. So they have the same architecture and they have the same function as the pore of all complex potassium channels, with the exception that they are very small. So they are not 600 amino acids big, they are only 80 amino acids big. And if somebody has a sort of a feeling for physics, this is exactly what you want if you want to understand something small and functional. So our work now is concentrated on understanding gating of ion channels. And we are now able to really put our finger on the mechanism, on the molecular movements and the physical-chemical interactions which happen in these proteins to make the opening and stochastic closing of these proteins. And unfortunately, there is a mistake in the image. It has been corrupted. What you would see there is that these channels, because of their small size, they're also ideal building blocks for engineering channels with new properties. So we've been creating, by putting together the ion channel from a virus and the photoreceptor from a plant, the blue light receptor from a plant, to make a blue light activated potassium channel. And this is, we have heard this before, this is a tool for optogenetics. And if the slide would not be corrupted, you would see that expressing this protein in the neuron is able to cause, by illuminating the protein with blue light, a long-lasting depolarization and the silencing of a firing neuron. So this is a short summary of the main research in our lab. Thank you.
Thank you very much, and it was really, really good to hear that how important it is for you to be member of the Academy of Europaea. So we really wish you all the best.
Thank you.
Thank you very much. So we have Irene Tracy from University of Oxford from the UK, and I think she is also here with us.
I am, thank you. And again, like others, it's just a real delight and thrill to be part of this fantastic academy. So thank you to the board for the kind election. My name is Irene Tracy. I'm based at the University of Oxford. I'm a neuroscientist with a focus on using neuroimaging tools to understand human pain, both as it's everyday, the normal everyday pain that we experience, that is our protective alarm system, but also as it goes wrong in the terrible medical condition of persistent chronic pain, which is one of the largest medical health problems we now have in the developed world. So if we go to the next slide, there's just a brief summary of our approach, which is to use these tools to go behind the scenes of, and if you just click one more time, there should be one more picture. That would be great. So the problem and challenge of persistent chronic pain is understanding what's going on behind the subjective report. It is a perception that emerges through brain processes, and the relationship between the injury to the actual perception is a highly non-linear, complex one. And that gap between the injury and the perception is where there can be many mechanisms that can go wrong and can contribute to both amplifying the pain, making it much worse, also turning it down, but also altering what parts of the brain those signals are allowed access to, and very much changing the quality of the experience. So it's very hard to know and understand somebody's pain based on looking at the injury, because of this journey from the injury to the brain, having these points where signals can be turned up, they can be turned down, and then ultimately where it emerges through brain mechanisms, there can be many different networks that can become active. So I've spent, and my team, we've spent 20 years trying to fill that gap between the injury and the perception, to uncover the mechanisms that contribute to the exacerbation, the maintenance, the persistence, the vulnerability, the amplification or the attenuation of these pain states, and to understand where these amplifiers are. I'm not sure if you can click, and there should be one more picture. If not, there it is. So again, from the periphery through to the spinal cord, through to the brain, we now have a really good understanding of the mechanisms that again can alter and contribute to that non-linearity between injury and perception. And these give us new opportunities to target, as I say, one of the largest medical health problems. 1 in 5 suffer with persistent chronic pain, and afford new ways that we can potentially monitor people's pain from a more objective perspective, as well as their capturing state. So again, apart from that, like many others, I have other sort of roles in terms of, again, contributing to neuroscience. Notably, just coming up, I will be taking over the presidency of the FENS organization, the Federation of European Neuroscience Societies. So really delighted to be part of this European family. Very honored to be elected, and thank you again. And I look forward to interacting with many of you in the future.
Thank you very much indeed. And maybe I ask you a silly question, because I'm absolutely not in the field, but in a pain as a multi-dimensional problem, of course, there are the sensualization of pain, like I feel the pain, and the other is about what the other problems the whole signaling mechanisms cause for us. So are they always associated with each other? So, or sometimes maybe I decrease the pain, but all the other negative effects is still on, or vice versa, or they are always associated?
It's a great question. So if I understand your question, you're asking if the pain intensity goes up from moderate to strong, unpleasantness go up from mildly unpleasant to very unpleasant. And normally, you're right, they always go in the same direction. but sometimes you can flip them and they can go opposite. So in, say, hypnotic states or in states of where you change the subjective value of pain, you might actually like the pain, you might find it pleasant, because the value of it you've altered in terms of reappraising the meaning of it. The analogy I'd often use is as an athlete who's maybe done an extensive training, say training for a marathon, and the day after their muscles are aching and their joints are aching. So this is pain, this is signaling tissue damage and repair, but they interpret it as something positive. They run, they're making their target, and that's a classic form of reappraisal, very what we call high brain. And again, we've done experiments simulating that. How is it that you can hedonically change the value of pain and actually flip it and make it pleasant, yet still know it hurts you? And then you can work out the reward mechanisms in the brain that are doing that. So those are the types of experiments we've done to understand all these weird different types of perceptions of pain that one is able to have in your everyday life, but also can go wrong in the clinical setting too.
Thank you very much indeed. So we have István Ulbert as the next a member of Academy of Europaea from the Research Center for Natural Sciences from Hungary. We have Basil van Engelen from Netherlands, from Radboud University Medical Center. We have Anna Widerholm from Karolinska Institute from Sweden, who is also a new member of the Physiology and Neuroscience section. And now I think we go for the next one, to the Organismic and Evolutionary biology section, which is led by Christophe Debaud. The first member of it, Zoltán Barta from the University of Debrecen, Hungary. And we have Michael Bruford from Cardiff University from UK. We have Austin Bird from the Imperial College London, UK, and I think he is with us, and he shared his actually slides with us.
Hello, yes. Thank you for the honor of being elected member to the Academia. So yeah, I'm an evolutionary geneticist by training. If we can just go to the next slide. Should be there, hopefully. So, so evolutionary genetics is my background, and the specific context in which I've been working for the last 15 years or so is to develop new methods genetic methods for controlling the vectors of disease, mosquitoes and other vectors of malaria and other diseases, and other pests. And so the mainstay, as you all know, for controlling pests is chemical control, insecticides, pesticides, rodenticides, etc. And those have been very good, but often they're not always enough. And we've got problems of resistance evolving. They can be expensive, and they're not always as targeted and specific as one would want. So we're interested in taking the modern methods of molecular biology and see to what extent we can use them in applied population biology for controlling harmful species. The main context focusing on is using so-called gene drive. So that's where you design a genetic construct that will be inherited at a greater than Mendelian rate. So instead of, if you're heterozygous, instead of getting 50% of the progeny, it'll get to 95 or 99% of the progeny. So a gene like that can spread through a population, even if it's causing some harm to the organisms causing it. So the idea is to picture there on the left, one strategy for doing this would be to take a nuclease, such as CRISPR-based nuclease, put it inside a fertility gene of your pest species, a gene that's necessary for fertility, where the knockout is recessive. And so then if that nuclease gene is expressed in the germline when the organism is making its sperm and its eggs, then it can cleave, and it recognizes the intact gene, then it can cleave that intact gene, get copied across, and so convert the heterozygote to the homozygote, and then get to all the progeny instead of 50%. And so if you do just very simple population models, you can release, 0.1% of the population carrying that construct. And then that will spread over successive generations. And as it does so, you'll be creating a population-wide knockout of that female fertility gene. And so the population then will go down. So that you can see as a potentially anyway, a very powerful, efficient way of controlling target species. Sometimes indeed that's too powerful. It's more powerful than you want. You only want, for example, to control a species in one location and not in another. And so we've also been working on developing ideas for CRISPR-based strategies for so-called self-limiting strategies, where you can release this construct and it would control the population in the location, but not spread outside of that location. And again, we've got designs that, at least in principle, ought to be like 100 times more efficient than the traditional sterile insect technique, which is just inundating a population with sterile males. So we've been working about strategizing on how different constructs could be used for different purposes. And then in parallel, I've been leading a multidisciplinary team that's specifically targeting Anopheles gambiae, the primary vector for malaria in sub-Saharan Africa. And so that's a team of members from 11 institutions, 4 of them in Africa and 7 outside of Africa. That's this Target Malaria Research Alliance. And so that takes the rest of my waking hours. So very, very happy to be part of this. And obviously this has policy implications, as was alluded to earlier. These are GM organisms for which the public are very interested. And so happy to be joining Academia because of their policy interests as well.
Thank you very much indeed. What do you think, how far we are from the therapy of malaria to control, and what actions should we do for that?
Well, so through collaborations with the lab of Andrea Crisanti, also here at Imperial, we've been able to eliminate small cage populations of mosquitoes, seed a population, then over successive generations, the gene spreads of its own accord and then collapses the population. We have done some non-gene drive releases in Africa of genetically modified mosquitoes, but just very small scale, which we will gradually be increasing the scale of. And I don't know if 5 or 6 years from now, hopefully we will be in a position to submit a dossier asking for permission to do a gene drive release. So that's the kind of time scale we think we'll be ready to have the information necessary for people to make an assessment.
Okay, thank you very much indeed. So the next member is Pedro Giordano from Spain, who is also a new member of Academy Europaea. We have a foreign member, again, Le Kang from Chinese Academy of Sciences from China, who is working in the ecological genomics. We have another member from France, from Purificación Lopez Garcia, who is also working with microbial evolution. We have a new member from the UK, from the University of Bristol, Jane Mammoth, who is working with community ecology. We have Ben Sheldon from the University of Oxford, from the UK, working with animal behavior. We have Kostas Triantis from Greece, from Athens, working on ecology and biogeography. We have now a new member from Singapore, who is actually, I think, with us, David Wardleb from the Technology University. I don't know whether he is here.
Yep.
So thank you for being with us. and please share your welcome and research with us.
Thank you very much, and thanks very much also for the election to the Academy. It's a real pleasure. So I'm currently based in Singapore. It's the Nanyang Technological Institute, University. I've been there for about 4.5 years, but I've spent the majority of my professional career in both New Zealand and in Sweden, where I still have really strong affiliations. My field is in terrestrial ecology. Specifically, it focuses on the linkage between above ground organisms, the plants and animals that we see above ground, and the organisms that we see in the soil, and how they interact to drive the functioning of systems. And I'm also interested in how these interactions are influenced by global change phenomena, such as those driven by our human activity. Next slide, please. Could you skip this? Yeah, so if you look at the center diagram, that represents the conceptual framework that my colleagues and myself look at with regard to this type of work. It recognizes that the plants and animals that we see above ground influence the organisms we see in the soil, both the decomposers, as well as those organisms more tightly linked with plant roots. And these organisms in turn interact to influence the plant and animal communities that we see above ground. So we apply this approach to a variety of different situations and systems around the world, as is shown by the various diagrams surrounding the central panel. For example, as I showed panel A, we do work looking at invasive animals in new environments and how they drive the functioning of ecosystems. These are invasive animals that are introduced to areas where they didn't previously exist, such as deer, and areas where deer didn't exist. We observe what happens when these invaders are brought to a new environment. Panel B reflects work we do on oceanic islands. We have islands with different levels of invasion by different sorts of organisms, and we see how this influences the way the system functions. With regard to panel C, we have a particular interest in fire. Fires are becoming more frequent in many parts of the world as a direct result of climate change and global warming. And panels D and E represent model systems that we use in northern Sweden based on islands, which differ in fire regimes. So different islands burn with different frequencies and have different fire histories. We compare different islands as model systems to see how fire history affects the way these islands function. We also work in the tropics in panel F. This is relates to work we do in Southeast Asia, Indonesia, Malaysia, and Brunei, where the forest area has been historically under pristine and primeval peatland forest. The forest has been cleared to make way for oil palm plantations. There's significant burning going on, which creates huge environmental damage, and also significant human health problems through smoke haze that crash respiratory issues. Our work is involved in trying to understand these systems and how to restore them back to something which was a bit like what they used to be before they were degraded. And with regard to panels G and H, this is work we do in the Swedish Arctic. It's an area that is disproportionately affected by climate change, and we're interested in how climate change affects the way these systems are going to function over the coming few decades. Now, for me, the highlight for this type of work is not just the interesting places we get to work in and the questions we get to address, but also with the tremendous bunch of collaborators that I've worked with on these problems over the past 30 years or so. And this includes the approximately 60 PhD students and postdocs that have come through our lab, through whom I've learned an enormous amount from, and contributed to many of the ideas that I've been working on myself. And now we're based in universities in 16 different countries and in 5 continents, where I feel they'll be part of the next generation of excellent scientists doing interesting things in the future. Thank you very much.
Thank you very much indeed. I just wonder how much do you need to travel actually to do this scientific activity, and how COVID-19 actually affected in your work?
Yes, there is quite, some of our work does involve quite some traveling because we're interested in global questions. I mean, we have studies, for example, looking at mountains around the world. So we send postdocs around to these different places or have local collaborators working in them. I would like to perhaps go to these places a bit more often myself, but obviously there's constraints with being in a university that have to be here sometimes as well. COVID has interfered with some of our work to varying degrees. In some cases, we've had local collaborators that can do all the work out there for us. We're very grateful to them. We have been able to do a small amount of travel. I have been able to actually travel to Brunei to do field work there because there are reciprocal travel arrangements between Singapore and Brunei. But yes, our wings are very much clipped and it's very much constrained what we can do. but we do what we can.
Okay, thank you very much indeed. It was very, very interesting to hear your presentations. Thank you very much. So now we go to the clinical and veterinary science section led by Stephen Holgate from Southampton, and let's look at the new members. We have Peter Ebbe from Denmark, who is an epidemiologist and working with the infection diseases. We have Arturo Anadon from Madrid, from Spain. We have Christine Stabelman from the University of Southern Denmark. From Denmark, of course, we have Thierry Calandra from Lausanne University Hospital, working with innate immunity. We have Timothy Cox from the University of Cambridge, who is working in human biochemical genetics. We have Inger Ekman from the University of Gothenburg from Sweden. We have Laura Ederkin Fleming from the European Centre for Environmental and Human Health from the UK. We have Dawn Sylvia Freshwater from the University of Leeds. From UK, we have Henk-Jan Guchelar from the Leiden University Medical Center from the Netherlands. We have Tracy Hassel from the University of Manchester from UK, working with the lung infection and inflammation. We have Alan David Irvine from the Trinity College Dublin from Ireland as a dermatologist. He's also very welcome. We have Frank Keeley from the Imperial College London from the UK. We have Gianluigi Lenzi from the Università di Roma from Italy concerning nuclear medicine. We have Karina Lovell from the University of Manchester from the UK. We have Mariam Fleur Moffat from the Imperial College London from the UK. We have Iris Nachtegaal from the Radboud University Medical Center from the Netherlands, working in gastrointestinal pathology. We have Anna-Marie Rafferty from the King's College London, UK. I don't know whether she is here still with us.
Yes, I am. Yes, I am. Yeah.
So please, if you can maybe say a couple of words about your research, we would very interested in hearing you.
Thank you so much for the honour of being elected, first and foremost, to the Academia. It's a great privilege. I'm going to follow, I think it was Frank Stewart, but reverse his comment, saying, you know, stick to a problem that you hope will not be solved in your lifetime. And I'm very interested and exercised by the global nursing shortage, and I sincerely hope that will be solved during my lifetime. And much of the research that I've had the privilege of being involved in, it's been team research like everyone, has tried to take little bite-sized chunks at that challenge. First of all, looking at the numbers of nurses and nurse staffing ratios in relation to patient, but also nurse outcomes, the costs and consequences of this global shortage. We are dealing with 20 million nurses missing from the global workforce, and that is nothing based on demand. It's only those positions which we think are actually funded, and the data sources for nurse staffing are very, very poor in many instances. So it's partly a plea. I work across history. I'm a historian as well. In policy and health services research, I think they all fit together quite well in analytically and in politicizing and trying to raise the profile of this issue up the agenda, because basically if we don't have enough nurses in any sector, we will not have resilient health systems, and we will not have health systems at all. So very open to collaboration from colleagues, and I'm very excited to be part of this stellar academy, and I've learned lots today. So thank you so much for all of those contributions from our esteemed colleagues. Thank you.
Thank you very much, and I am so happy that Stephen Holgate also worked very, very effectively concerning bringing nursing research to this section of clinical sciences, because I think during COVID-19, it was very clear that not the number of mechanical ventilators were the biggest shortage concerning the treatment, but the nurses. So I think also like this is the field where might be Academy Europaea can also be part of highlighting this importance. And as I mentioned earlier as well, and to anybody here, be active, send proposals to the leading committees, and maybe if there are teams, research teams forming, I think this is what Academy of Europaea about. Thank you very much.
Thank you.
And I think we also missed somebody from the list. I don't know whether Patricia Holm is here with us still.
I'm here, yes.
Yes,
hello.
we are very sorry to miss you.
No problem. I thank you very much for electing me, and I'm very glad to be with you today, at least online. So my name is Patricia Holm. I'm professor for ecology at University of Basel in Switzerland, and my background is in biology. I'm an environmental biologist and doing research on invasion biology, mainly in aquatic systems. I also work on microplastics in the River Rhine, as well as in some oceans, like the Southern Ocean. We are looking for the occurrence, distribution, but also for the effects of microplastics on the Antarctic and on the Rhine ecosystems. In invasion biology, I'm mainly also working with fish, invasive fish invading our rivers and lakes. And in addition, I'm active in cetacean research and especially interested at the interface between science and policy, doing a lot of interdisciplinary studies and also transdisciplinary studies with stakeholders and participatory research approaches. And we have the Master on Sustainable Development at our university in Basel, which is really completely interdisciplinary from the very beginning, working with scientists and researchers, lecturers from the different disciplines to educate our next generation for sustainability. That's in short my profile. Thank you very much for your interest.
Thank you very much, and I think it's really very hard to find the channels to policymakers actually from the scientific approach. So how you can reach them? What kind of channels do you use to reach the decision makers?
Yeah, I think it's pretty much easy in Switzerland because we are such a small country and we have a direct democracy, as you might know. So you are very easy in close contact, but I'm also interested in the international big organizations like the International Whaling Commission, where I'm chairing one of the big subcommissions in the scientific committee, namely on environmental impacts. I'm also a delegate for Switzerland for the UNEP, working on microplastics in the scientific advisory board. So I try really on different levels to interact with the policymakers and the decision makers. That's really my passion.
Oh, thank you very much indeed. Very hard work for sure. Thank you. I think I also missed Tom Cox, who is also present, and I think I just jumped over him. Are you here, Tom?
Yes, I am. I'm
Thank so sorry.
No, it's perfectly okay. I've enjoyed listening to all the other talks. I've been quite inspired, actually, not only within our own system, but a huge thanks to Professor Stephen Coldate and Balázs Gulyás, actually, for sort of putting me forward, and to the council for electing me. It is a huge honor, and I feel very much European, particularly at this time, with many collaborators and colleagues for many years in Europe. I'm a physician of quite a long standing and 32 years in Cambridge, and all of us, of course, interested in the future, and the future together with European scientists and others too. This is not just like getting a piece of your money, it's a question of being colleagues and with a common thinking. My research is in rare diseases, which are very revealing, and I can quickly mention them. They go from strength, in other words, iron, metabolism. They went to sweet things, fructose, and finding causes for problems with fructose in the diet that were really genetic and very fascinating and revealing. And then sourness, the acidic pH of the lysosome, and the diseases of the lysosome, particularly sphingolipid diseases, and actually the interaction with the immune system and innate immunity, which we've made a recent finding in, and which I hope will be published. But it relates very much to some of the other talks, which is lovely to hear. Valerie from Cardiff and Lars Nietzsche, for example, already colleagues I can think we can do some things with. But anyway, that's my research. Well, everyone likes to talk about their research, which I'd be happy to do. But I'd like also to talk about the future, possibly something that has not come up so far. And that is the idea of bringing science into medicine, because science owes a huge debt to medicine, as you know, Chairman. Because what you reveal from particularly the rare disorders, a buzzword today, the inborn errors and things, of what you learn anyway, that the people who drive innovation are almost always people who've seen the diseases and know them somehow. They may not have made the primary scientific discoveries, sometimes they have, but they drive the application and translation. And the sort of people that do that are those now increasingly with a really authentic clinical research education, and the research, true research, is the difficult thing to get. So in Cambridge, I did have the pleasure 30 years ago of starting the MB PhD program, which was the first in Europe, in the UK, a rare one. There are various variants in Europe, particularly in Scandinavia and Sweden, particularly, and also the other Germanic tradition of Scandinavia. But truth is, this was an integrated program, which was thought to be for the very rare and the terribly intelligent. It's about 5 to 10%. Many of them are women who choose to do this, and they stay with this tough program where they are integrated with a research project for their own after they've seen patients, but within their clinical course of the, shall we say, the dark arts of clinical medicine and the understanding of human relations and delivering of therapy and negotiation with the complex organism of the human. And so it's just something that I think I feel very strongly about. I'm not forcing it on anyone, but there has been a great deal of interest, I know, by the Academy in this element of the future. In other words, growing clinical scientists, clinical academics who remain clinically affiliated and sometimes clinically active into a proper doctoral research program, which they then use to build their careers. So it's something I feel very strongly about. I can just hint about it now. I think it is, to a certain extent, the future, well established in the United States. Many of the medically qualified people with a perspective in the Academy will have got a medical qualification, even if they're not really using it. But when we look back after 30 years of investment in this program, we've now got about, follow up on about, I don't know, 200 or so people. And there are one or two other programs, one in Manchester, UCL, and a few other universities still thinking about it within the context of the changing educational system for medics, as they say. The fact is, many of them are clinical, but many of them also are clinical academics who have independent research funds prosecuting their own areas of research at a very high level. And in that sense, it has clearly been a very, not the only, but a very, very useful pathway for of success to recruit. There's a shortage of nurses, but there's a shortage of adequately trained, critically able scientists actually at the practice of the new molecular sciences and everything else, the big data and so on, within clinical medicine. So I just wanted to say that little buzz of one of my visions personally for the future, and I'm sure it's something we can share with colleagues and have a dialogue and discuss in the Academy, which I'd like to do very much. So I'm extremely grateful to Steve Holgate for tolerating an application somewhere along the line from Balash on nominating me from Professor Guyette in Singapore. And thank you very much for joining me, this very special club, as someone on a small island. Thank you.
Tom, thank you very much indeed, and what you have done not only in the research field but also for healthcare, it's excellent, and it's very nice meeting you here, especially we also try to make a huge effort about giving science education for practitioners, especially at the young age, because I think having MD PhD programs and also to make residency PhD programs together, I think this is a must. Otherwise, we will not have a good quality healthcare for sure. So thank you very much, Tom.
Thank you very much indeed. Thank you for inviting me again. And to a warm welcome. Thank you.
Thank you very much. So for the next member is Roberto Rodriguez Roisin from Barcelona, from Spain. We have also an immunologist from Hungary, Zoltán Szekonec, from the University of Debrecen, from Hungary. We have Marianne van Haag from Karolinska Institute from Sweden, and as far as I understood from the office correctly, she has a short video to share, so I stopped sharing and I asked Phil and Juliet to show the video.
Dear colleagues, my name is Marianne van Haag and I'm honored to be part of the Academia Europaea. I am Professor of Clinical Immunology at Karolinska Institute, Stockholm, Sweden, and Senior Consultant Clinical Immunologist at the Department of Clinical Immunology and Transfusion Medicine, Karolinska University Hospital. I have done my MD and PhD training at Karolinska Institute. I have several commissions of trust. I am board member of several scientific foundations, in addition to being editorial board member of several prestigious journals in the field of allergy. My research area is allergic diseases, which are one of the most common chronic inflammatory diseases in Europe. My aim is to acquire novel mechanistic and clinical insights into severe allergic disease and to obtain biomarkers for risk assessment, prognosis and prevention, as well as to obtain safe and efficient therapies. I use a molecular-based allergen approach to obtain the goals. I have a special interest in severe food allergy and above all in mammalian meat allergy, a novel form of food allergy where patients experience delayed severe allergic reactions after mammalian meat intake due to IgE antibodies against the carbohydrate alpha-gal. The disease is associated with tick bites. I am also interested in respiratory allergy to obtain biomarkers for prediction of allergic disease. I am partner of the BAMSE team, and BAMSE is a large birth cohort where the children have been followed from childhood up to young adulthood. Here we are elucidating biomarkers, allergen molecules for risk assessment and prediction. I am partner of a newly started large EU project with the aim to elucidate how micro and nanoplastics interact with allergens and the immune system and the risk of allergic disease. I have many international collaborations and more than 380 original articles, reviews, editorials, and book chapters, and with an H index of 76. I am very much looking forward to being part of Academia Europaea. Thank you very much for your attention.
So unfortunately, here we cannot have questions. So, however, we can move forward. And I think it's one of my greatest privilege tonight or today to introduce László Vécsey, who I have been working with for more than 20 years. And he's not only a brilliant neuroscientist, but had lots of activities as official for science, for youth academies. He was also a member of and the chair of the medical section at the Hungarian Academy of Sciences. And I think his involvement in science also in Hungary, it's not questionable that he must be a member of the Academy of Europaea. And I would like to ask László, as a scientist and also my friend, to introduce yourself.
Thank you very much for electing me the member of the Academy. I think it's a really great pleasure for me. Concerning my workplace, I am working at the University of Szeged, the Albert Szent Györgyi Medical Center at the Department of Neurology and the Neuroscience Research Group. Our research is focused mainly on the role of the kynurenine or kynurenine system in different models of neurological disorders. So what we did in multiple sclerosis, in Parkinson's disease, in migraine and the stroke, we investigated the role of the kynurenines. And we are very happy that we have good organic chemist collaborators, and they synthesized us a couple of analogs, many of the analogs of kynurenic acid, and we have some kind of drug research program of at the clinic. So basically, I am a clinical neurologist, so therefore we are focusing on clinics. And what we did during the last periods, we investigated, for example, the neurofilaments, the kynurenins in multiple sclerosis, and also we investigated several biomarkers in migraine, and also we studied the same things in stroke, not just from the molecular point of view, but also the genetical alterations of the genes who are responsible for different kynurenine pathways. So in conclusion, we are mainly focused on molecular mechanisms of neurological disorders, but also we have a very good neuroimaging research group too. And we have some studies in multiple sclerosis and also in stroke. Concerning my activities, as it was mentioned, I was the chair of the vice chair and after chair of the medical class of our academy, and I was a representative for a couple of years of the Federation of European Medical Academies, which is also was a very important responsibility for me. Thank you very much again for electing me the member of the Academy. It is really a great pleasure for me. Thank you very much.
Thank you very much, László. And I just 1 week ago heard the presentation, one of your talents concerning this molecular biomarkers of multiple sclerosis in the radiology MRI measurements. So what do you think, when will we be able to predict or actually the probability of the onset of multiple sclerosis?
Thank you very much. I think this is not just to be polite, but really this is a very important question, because at the early stage of multiple sclerosis, we are just seeing after the clinical stage of the patient. We have MRI data, but there are big differences between the clinical future of these patients. So, for example, if somebody has, let's say, some kind of high quinolinic acid, which is an excitotoxin in the blood of patients, compared with those patients who has a low quinolinic acid, probably those patients who has a more excitotoxic activity, they should be treated more aggressively. And I think we are just in the stage to introduce neurofilament in the everyday practice, which is related to the neuroaxonal damage. So I think when we see the patients, we just see the tip of the iceberg. But if we have the chance to see the molecular biomarkers, we have a much broader knowledge about what's going on in the disease, and we have a better chance to find the best treatment strategies of these patients.
Thank you very much indeed, and it was my pleasure to introduce you as a new member of Academy of Sciences Europaea. Thank you very much. And finally, we have Maria Jasdan-Baks from Leiden University Medical Center from the Netherlands, but I think she might have to leave and cannot be with us any longer. So I think I can finally say again that welcome to the Academy of European new members. I don't know whether we missed anybody or if anybody would like to say a couple of words. Or maybe we have somebody from the other section who came here from section B.
Yes, you have.
Hi, Yark. Thank you very much for coming over to our class, and please also introduce yourself.
Thank you. First of all, I have to apologize for being in the wrong class. My class meeting was last week and I was not able to attend. And then for some reason, I was invited into this section. And now the list of my activities or interests which I have been involved in the last 50 years is here, and you can see that some bullet points are probably familiar even for you. So, and to justify this honor to be invited, I selected 3 keywords. Can I have the next slide, please? I selected next slide, please. Yeah. I selected 3 keywords: chemical kinetics, reaction mechanism, and structure-activity relationships, and formulated few sentences about my research interests since 1979, and this interest is still alive. And the main outcome of this research was that efficient ligands, which interact with receptors, but also biological molecules, this interaction cannot be described as a one-step binding equilibrium or dissociation equilibrium, but includes at least 2 steps or even more. What's the meaning? The meaning is that now, instead of working in one-dimensional data space, we have to go over to 3-dimensional data space. And this opens additional possibilities. So we can ask how are interacting agonists and antagonists, and how are interacting partial agonist and partial antagonist and allosteric ligands. So the 3-dimensional database is extremely complicated, but as we have a toolbox for experimental investigation into this problem, we think that at least in future, this will be quite a promising approach. So that's briefly everything I would like to tell. Thanks.
Thank you very much, and I must confirm you that you are in a very good section and very good class for sure. I think science is definitely multi and interdisciplinary, and as you just highlighted that the pharmacokinetics and the different chemicals working on the different receptors are not only for class B, but also class C. I think one of the biggest aim of Academy Europaea to build bridges, as it's in the motto, between not only scientists and public, but among scientists and between classes. So thank you very much for sharing your history and your work with class C.
Thank you.
Thank you. I don't know whether is there anybody else who would like to say a couple of words, or maybe we missed somebody. Just raise your hands. I think it's really a good company, and I really enjoyed myself of sharing these new members. However, if you don't have currently any other words, I would like to hand over to the microphone back to our president to say his final words. And now, Sir, the microphone is yours, and we are very much looking forward to your words.
Thank you very much, Peter, for sharing this very inspiring session, and of course also for all your very insightful and to the point questions. I think all of you who have attended this session will agree with me that this has been a very exciting meeting of Class C. Of course, a fantastic presentation by Katalin Kariko, but also it was very wonderful to see the warm welcome to our new members, and also to see their enthusiasm and their passion for science. People with a wide spectrum of backgrounds and scientific fields, and clearly all very active scientists. As I mentioned in my introduction earlier this afternoon, our academy is all about people, and we count on you for the future of our academy. Let me repeat just 3 examples where we really need you. First of all, of course, the issue of the scientific advice for policy, and many of you have mentioned this in their short introductions. We need you in the experts working groups for SAPIA. We need you also in bridging to the young researchers in your role as a role model, but also in bridging to the Young Academy of Europe, which is affiliated with Academia Europaea. And of course, we count on you for the activities in Class C, but also in interacting with the other classes of our academy. Coming Wednesday and Thursday, a stellar program is waiting for you for our annual conference. Please also join us on Thursday morning on the AGM. Finally, big thank you to the organizers, Eva and Peter. Again, you made here the difference. And of course, also big thank you to the technical support offered by the Cardiff hub of our academy. Again, very warm welcome to our new members, and wishing them much inspiration to their membership. Thanks for joining us.